Boa tarde a todos. Nós retornamos, então, às atividades com a sequência de palestras, agora no período da tarde. A primeira palestra será da professora Andrea Taylor. Vou passar a palavra ao professor Callum para que possa apresentá-la.
Okay. Thank you. Thank you, Anna. Welcome back, everybody. It's my pleasure to introduce Andrea Taylor. Dr. Taylor and I have worked together for a long time, modelling and studying feeding biomechanics in primates. She's a world expert on muscle architecture, and she's going to talk to us about functional evolutionary relationships between jaw muscle fibre architecture, phenotype, and feeding behaviour. Thanks, Andrea.
Thank you, Callum. Okay, well, I just want to say thank you to the organizers for putting this really wonderful conference together. I've already learned a lot from people that I work with every day, and I'm sure we're going to hear some really interesting talks between today and tomorrow, so thank you. So I'm just going to start by saying that, like most of the people who have talked already this morning and some of the people that you're going to hear tomorrow, my research interest is really focused on what drives the evolution of the feeding system morphology. So what factors drive that feeding system morphology? And, you know, we have heard a lot about bite performance today, biting and bite performance. And clearly for humans, biting is a key performance variable. It has an impact on quality of life. It enables us to feed on a variety of different foods. You can see that we face challenges depending on what kind of food items we're putting in our mouths and how large our mouths can open. And you know, young humans have to face those same challenges with smaller mouths. And in fact, in humans, we see that biting is not just a feeding behavior. This is an Inuit woman who's using her teeth and generating quite a bit of force to soften leather, basically to soften skin. And so I think we've heard a lot about bite performance today, and this is an important performance variable for humans as it is for non-human primates as well. We talked a little bit this morning about biting with non-human primates and the importance for non-human primates. And just like humans, primates put a variety of different food items. They have to ingest a variety of different food items and access them in different ways. You can see that we have different primates here who are all trying to get into a variety of items. Some are using incisors, some are placing them further posterior on their mouth. And like humans, I just want to point out that they're using, some of these animals are generating a fairly large gape to do this behavior. And Pepe did a very nice job this morning of talking a little bit about trade-offs between facilitating a large muscle and bite forces on the one hand and facilitating muscle stretch and wide jaw gapes on the other. So we know a lot about biting performance, but another performance variable that we really didn't start to think about, I don't think, that we did a lot of work on this until the late 90s, early 2000s. Certainly there were some people in the 70s and 80s who were talking about gape, and there were a couple of papers that were put out there, but we really didn't start to think about the importance of gape until the late 90s, early 2000s. And in this particular case, I'm talking here about gape performance in a behavior that's entirely unrelated to feeding. So we have bite force and bite performance. We have gape related to feeding. And then we actually have gape that's completely unrelated to feeding. But it's the same system that we use to feed. And so here we have a very classic group of old world primates that open their mouths widely to basically display sort of an aggressive way against other males when they're competing for females. And so sometimes they're generating these wide mouths to show their canines and to show what they have, and maybe they don't really want to engage in an agonistic biting behavior, but sometimes they do have to bite. And when they bite, they have to open their mouth wide enough to get their mouth around the neck or the limb or whatever it is that they're going after. So, but this does not have anything to do with feeding. So I'm going to talk about the relationship between jaw muscle fiber architecture and feeding behavior. and biting performance and gape performance. And I'm going to talk about bite performance and biting in the context of feeding. And we're going to look at some architectural work in that context. I'm going to look at some work that we've done on wide mouth opening and feeding performance. And then we're also going to look at some architectural work in primates that generate wide mouth opening for non-feeding behaviors. And we're going to talk a little bit about some of the trade-offs that these animals face. And then I'm also going to talk about jaw muscle fiber phenotype. This is some work that I've started a couple of years ago. So we're going to talk about combining fiber architecture and fiber phenotype, and how fiber phenotype might fine-tune the jaw muscles for specific motor tasks. All right, so heard a lot this morning about tufted capuchins. They're very well studied in the wild. Their behavior is very well documented. Their feeding behavior and diet is very well documented, and they have become an useful lab animal. And this is work that was done out of Callum's lab. I'm going to come back to the beginning of this. I really want you to see that you've heard about this behavior this morning. They are hard object feeders. They are very capable of feeding on objects that you or I would not want to put in our mouths and try to open. We would use a nutcracker. If we tried to do this ourselves, we would break our teeth or we would hurt our jaws. So I'm going to show you this again. This is a capuchin. This is a Shemp. Whoops. And he's like the Terminator. He has no problem breaking through these Brazil nuts. They're in their shell, so they're very hard nuts. And he's putting them one right after the other. He went through about 15 or 20 of these with no problem whatsoever. And you can hear him, right, cracking through the nut. And you can see he's maximizing, he's smart enough. He's maximizing his leverage there. He's opening his mouth widely. He wants to put that on the back teeth so that he can generate a pretty good force back there on the post-canine teeth. All right, so when I started wanting to look at fiber architecture and its relationship to feeding system morphology, I decided I really wanted to start with extreme behaviors, because if you can't find a signal in extreme behaviors, then game over. You might as well just go home. So I said, well, let's start with some extreme feeders. And capuchins are one. I'm going to talk about another hard object feeder, sooty mangabeys, and I'm also going to talk about a species that opens its mouth widely. That's an extreme behavior. They are tree gougers. And you saw a picture of the tree gougers this morning in Pepe's talk. So we started with hard object feeding tufted capuchins, and we said, well, let's look at their architecture and compare them to non-hard object feeders that are also closely related. They're untufted capuchins. And let's take a look at their architecture. We would predict, given their feeding behavior, that they would have the capacity to generate relatively large muscle and bite force. And so we would be looking for architectural features that would facilitate that behavior. So what did we find? I just want to start, before I show you some data, by talking about fiber architecture and what I mean by fiber architecture. And it's really the internal arrangement of fibers relative to the force-generating axis of the muscle. And Pepe did a nice job this morning talking about internal configuration. We're talking about fiber length and the orientation of the fibers relative to the force-generating axis of the muscle. Some are more pennate than others. This is a highly pennate muscle. This is the superficial masseter. Temporalis is less pennate, but still pennate muscle. And fiber architecture, fiber length, muscle mass, pennation angle, those can get us to two important whole muscle variables. One is physiologic cross-sectional area, which tells us something about the whole muscle force, the force-generating capacity of a muscle. And the other is fiber length, which tells us something about muscle excursion or stretch, and by extension, contraction velocity. This is whole muscle now. All right. So we looked at physiologic cross-sectional area. We compared tufted capuchins to untufted capuchins. They differ in size, so we had to do scaling to adjust for the fact that they differ in size. So relative PCSA just means it's PCSA adjusted for size. And what we found is exactly what we predicted, which is that tufted capuchins have relatively larger cross-sectional areas, both for their superficial masseter and their temporalis. We don't see that difference in the deep masseter. Deep masseter is really not that important for generating large bite force. But we see it in the superficial masseter and the temporalis. So this looks like a good picture, and it looks like what we would expect, and it's a nice story. So we should expect all hard object feeders to have relatively large cross-sectional areas to facilitate muscle and bite force. I looked at another hard object feeder. This is a sooty mangabey. It is Circassibasatus. It's an old world monkey. Tufted capuchins are a new world monkey. Work coming out in the last couple of years now. Very excellent work from the field documenting very fine-grained behavior in males and females, adults, juveniles, and also quite a bit of comparative morphological work now. And I'll show you a video. This is Circassibasatus feeding in the wild. They feed on extremely hard and large seeds. I'll show you a picture of it downstream. They're very large, they're very hard. They're not interested in the shell, but they have to get through the shell. They're interested in a very small, oily nut that's really easy to bite. But to get to it, they have to get through a really complex structure. And you can hear it. These nuts, they litter the forest floor. They eat these nuts habitually all day long. They just sit on the forest floor, pick them up, put them in their mouth, break it, take another one, put it in their mouth, break it. Sometimes they pick one up, put it in their mouth, they don't like it for some reason, and they toss it. Interesting to find out why. All right. So same question, same expectation. We expected these sooty mangabeys to have architectural features of the jaw muscles that would facilitate generating large muscle and bite force. And so we compared these sooty mangabeys to a group of very closely related old world monkeys that are not specialized hard object feeders. Baboons and 2 species of macaques. And we found that sooty mangabeys do not systematically have relatively larger cross-sectional areas. Baboons, even when we scale for size, have larger muscle cross-sectional areas. They're not hard object feeders. Sometimes they have larger, sometimes the sooty mangabeys are larger than the macaques, not always. But we don't see a systematic pattern here where the hard object feeder differs in the predicted direction compared to the non-hard object feeders. We saw this result whether we used jaw length as an independent variable, because we can use jaw length as a load estimate for biting at the incisors. Sometimes they do bite at the incisors. We found the same result when we looked at an estimate for chewing or biting at the molars. Doesn't matter. They don't fit the expectation. All right. Keep that in the back of your mind. Those are extreme examples of hard object feeders. I wanted to look at the other side of the equation. I wanted to look at architectural, I wanted to look at an animal and a feeding behavior that requires generating relatively wide jaw gapes, to understand what their fiber architecture might look like and how that might contribute to their feeding behavior. This is a common marmoset. It's a tree gouger. In the wild, they anchor their upper dentition into the tree, right into the tree bark. They use their lower incisors to plane the bark. They are stimulating the flow of gums and saps. So the gums and saps are really easy. They just lick those things up. But they have to get through the substrate to release those gums and saps. Whoops. This was done in the lab of another collaborator of mine, Chris Vineyard at Neomed. And actually, this was done at Duke. He's now at Neomed. And you have to watch carefully because he'll block himself a little bit, but you're going to see where he's going to... There's gummy bears in this wood. They love gummy bears. Watch him. There. See it? Very wide mouth as he's using his lower incisors to plane. This is a behavior that clearly requires generating relatively wide jaw gapes. It's fitted to a force plate, so they were able to measure the bite forces that were associated with this behavior as well. So they measured gapes, bite forces, went into the field in the wild, observed them, measured their gapes in the wild. they are actively generating gapes that are approaching their maximum structural capacity for gait. So Pepe this morning did a nice job talking about the sweet spot on a length tension curve for muscle force, which is the plateau region of a length tension curve. Every muscle has a sweet spot for generating, for being in a position for optimal force. And if you're on the ascending limb of the length tension curve, the sarcomeres that make up that muscle are going to start to overlap more and more, and force is going to drop off. And if the muscles start to stretch on the descending limb of the length tension curve, those sarcomeres are going to start to separate, and without the optimal overlap, you're going to see a drop in force. And the animals I just showed you, the common marmosets, they're operating somewhere down here. So they are opening their mouths very widely. Those muscles are on stretch, and that's when they are planing this substrate. So we have a number of, we've done a lot of work on this animal, and we have some idea of how they're able to do this. It happens that even though they are on that descending limb of the length tension curve, they have some features that enable them to generate adequate bite force at that gait. But I can tell you that for a long time, this behavior was assumed to require relatively large muscle and bite force. And the data show that tree gouging doesn't actually require large bite force at all. It does require, however, a large jaw gait. So I said, well, let's compare architecture of these tree gougers to closely related non-gougers. We're looking at common marmosets up here. They have a sister pygmy marmoset. This is an extremely small primate, would fit in the whole primate would fit in your hand. And yet they generate the same behavior as the common marmosets. And we said, well, what would we expect? We would expect architectural features of the jaw muscles that would facilitate muscle stretch and the ability to generate a relatively wide jaw gait. All right, so we looked at fiber length here. This, again, these are all different size, so we scaled. Masseter is on the left here, temporalis is on the right, and the non-gouger is in the middle in all of these figures. And the common marmoset and the pygmy marmoset, really tiny primate, both of these animals have relatively longer fibers than the non-gouger. So again, looks like it fits a nice picture there, that they have features that facilitate that muscle stretch. We found that they do not have large physiologic cross-sectional areas, and it makes sense because this is not a high muscle bite force behavior. So in fact, the non-gougers here have relatively larger PCSAs compared to the gougers. And in this case, we found that if you look at increasing excursion on the x-axis and increasing force on the y-axis, that as you increase, go from increasing force to increasing excursion, you see that these marmosets are actually trading. They're trading. They're facing a trade-off, and they are not able to maximize their muscle force and their muscle excursion. So they do experience a trade-off here between force and excursion. Well, I'm going to come back to the capuchins that I talked about earlier, because Pepe this morning talked about different ways in which muscle can be reorganized to alter its architecture. One way to increase physiologic cross-sectional area, if you want to be economic about it, meaning you don't want to spend the energy to add more muscle mass, is you maintain the similar muscle mass, but you rearrange the internal architecture. If you want to increase the force, you're going to decrease your fiber length, and you're going to increase your angle of pennation, and you'll increase your force. But you don't always do that. You don't have to do that. You could add muscle mass. And in the case of the marmosets that I just showed you, they didn't add muscle mass. They underwent this trade-off. They relinquished some force. They were able to generate muscle stretch and wide jaw gapes, but it came at some expense to muscle force. But we didn't see that trade-off in our capuchin monkeys. This is what I showed you earlier. The tufted capuchins have relatively larger cross-sectional areas than the untufted capuchins, but it turns out that they did not sacrifice fiber length or pennation angle to achieve that configuration. They simply added more muscle mass. So they have relatively larger cross-sectional areas, but it's really not because they underwent an internal rearrangement of their architecture. They added muscle mass. It could be potentially expensive, but it does allow them to access resources that untufted capuchins cannot access. So we don't always see that trade-off. All right. So what about wide mouth opening unrelated to feeding performance? I showed you earlier we have a group of primates that are sort of quintessential display primates. They like to show off their canines. They engage in aggressive behaviors to compete for males. And to do this, they open their mouths extraordinarily widely. So that's a muscle that's on stretch. Right? Those muscles are on stretch. Now, this is a display behavior in which this animal is not actively biting, but they do open their mouths widely to actively bite if they need to. Yeah. Underwhelming compared to the mandrill. So we wanted to ask the question, well, what does the architecture of their muscles look like in these unusual primates? They're unusual for a number of reasons, but I showed you the sooty mangabey, which happens to be one of these primates, that engages actually in gape display behaviors, but also has to generate adequate bite force at a fairly wide jaw gape when they pop these saccoglottis seeds in their mouths. So we started by asking what males and females look like, because the behavior here that I'm showing you is really a male-dominated behavior, this gape display. Females sometimes display, but they don't have the canines, they don't have the facial configuration that the females have. So we wanted to say, well, let's start with males and females and ask whether we see a difference just between males and females of the same species in a species that engages in this gape display behavior. And right off the bat, you can see that these are dimorphic. Males are larger. They have larger muscles. They have much more projecting canines. They're a dimorphic species, and this is a behavior that we tend to see in dimorphic species. So we looked at the masseter. We looked at fiber length. We expected that males would have relatively longer fibers, architectural features that would facilitate producing those wide jaw gapes. And we did find that males have relatively longer fibers in their anterior masseter, which is quite stretched when they open their mouths widely. They also have relatively longer temporalis fibers compared to females. So in both cases, both muscles, males have the expected configuration. Did they trade? Did this configuration come at some expense to muscle force? And the answer is no. In fact, males and females have comparable cross-sectional areas for their superficial masseter, and in males have relatively larger temporalis cross-sectional areas. And so they do not undergo this, trade-off between force and excursion. What they do is effectively what capuchins do, which is that they've added muscle mass. So they've not changed the internal configuration of their architecture. Maybe this is just unique to macaques. So we went back and we looked at this in the sooty mangabeys that also gape display. They're an old world monkey. They're sexually dimorphic. We found the same pattern, longer fibers in males compared to females, both muscles. This is superficial masseter on the left. This is temporalis. I'm sorry, this is superficial masseter relative to jaw length. This is superficial masseter relative to condyle M1 length. It doesn't matter. And in fact, we went back and looked at this across a variety of sexually dimorphic old world monkeys. And this is simply a correlation here between jaw gape on the y-axis and superficial masseter fiber length on the x-axis. And what we find is that there's a very strong and significant correlation between gape and fiber length in males, but there's no correlation in females. And this makes sense because when you look at gape and canine size in these same species, gape and canine size are correlated in males, but not females. So here is an architectural configuration in a group of animals that impacts their feeding system morphology, but has absolutely nothing to do with feeding. And it really has something to do with reproductive success as it relates to competing for mates. But it's impacting the feeding system. So when you go back to this morning, you think about Pepe's talk where he showed that last slide about how complex it is when you're thinking about what drives the morphology of the system. It's very complicated, and it's not all about feeding. So just to summarize, some hard-to-object feeders have relatively large jaw muscle cross-sectional areas that facilitate generating relatively large bite force. Some don't. Some species experience architectural trade-offs between maximizing fiber length and cross-sectional area. Some don't. And some species are optimizing fiber length for non-feeding oral behaviors, in this case, aggressive gape display. So a picture that we started out thinking, this is great, we're getting exactly the signal that we were hoping to get, turns out to be a lot more complicated than we thought it would be when we first started out looking at common marmosets and tufted capuchins. So I'm going to set aside architecture, and I'm going to come now to some newer work that we've been doing on fiber phenotype of the jaw muscles. When we heard the talks this morning about modeling, the point that was made over and over again was how important the accuracy of the data is, that the inputs into the models for finite element modeling and any other modeling that we might be doing. And one of the kinds of data that we have been ignoring in these models is fiber type. So we've been assuming that all cross-sectional area, I've been making that assumption, and everybody who uses those data is using data that is assumed that the cross-sectional area of the muscle is comprised of the same fibers. But we know that's not true. So what do I mean by fiber phenotype? I'm talking about physiological characteristics of muscle fibers. The physiologic characteristics are things like endurance or fatigue resistance, contraction velocity, and the cost to tension. How much does it cost to recruit and contract the fibers? This is actually from one of our sooty mangabeys. This is a superficial masseter, and I'm going to show you a couple of slides of some of these data. I want to start by talking about fiber types, and in particular here, I'm interested in terms of the contractile proteins found in skeletal muscle. We're looking at the myosin heavy chain isoforms. These display the greatest diversity, and there are 10 myosin heavy chain isoforms. Some of these are so specialized I'm not even going to talk about them. I'm only going to talk about 6, and 4 of them are found in limb muscle, and 2 additional ones are found in jaw muscle that are not found in limb muscle. So right off the bat, we have 4 fiber types up here, 1 beta, 2A, 2X, and 2B. These are slower fiber types on the left here. So if you look on the left of this continuum, on the left-hand side, we have low velocity, high endurance, or high fatigue resistance. You can contract them over and over again. And they come at a low tension cost. And on the right-hand side, it's the complete opposite. This is a high velocity, but low endurance. They fatigue rapidly, and they come at a high tension cost. So 1 beta or type 1 fibers are on the left-hand side here, and then we have various different isoforms of type 2. And these are found in limb muscles and jaw muscles. But in jaw muscles, we also have additional myosin heavy chain isoforms. We have 1-alpha or alpha cardiac. They're the same that are found in your heart muscle. And 2M, masticatory myosin, a specialized myosin that's found only in jaw muscles. I want to say that humans do not express type 2M. The gene encoding this myosin is present, but it was inactivated by a frame-shifting mutation. And so we do not express type 2M in humans. So this is the full continuum that we see in jaw muscles. And again, starting on the left-hand side here, you can see the different properties of these fibers as we go from 1-beta, 1-alpha, all the way to type 2. And you notice that there are parentheses here, and that's because we also find a continuum of hybrid fibers, meaning fibers that co-express more than one myosin heavy chain isoform. So we find fibers that express 1-beta and 1-alpha together. We find fibers that express 1 and 2A together, and so on. We find hybrid fibers in limb muscles, but much lower in terms of prevalence than what we see in jaw muscles. Now, the continuum of this myosin heavy chain content has been linked, I don't want to say tightly, but it's linked to function of muscle fibers in some non-primate mammals. Largely in grazers and ruminants, in some marsupials, in some small mammals like rabbits, and in eutherian carnivores. And we can map these animals along this continuum, and what we can see here is that we have grazers, and grazers like sheep and cattle. They have largely a 1-beta, a slow phenotype. If you think about, they execute tens of thousands of chews a day. If you've ever watched a cow, they just chew all day long. Low amplitude, but they chew all day long. So they have a fiber type that allows them to chew all day long. It doesn't cost a lot. They don't have to generate a lot of power to do it, but they're fatigue resistant. So it's a good setup for them. These kangaroos here are also a slower phenotype, but not quite as slow. They have almost uniformly 1-alpha in their jaw muscles. So faster contraction time than 1-beta, but still at a low tension cost. They are grazers like these sheep, but they are not ruminants, so they don't ruminate. So the 1-alpha has been linked to giving them a little bit more efficiency of breakdown in their food because they're not ruminants. So it has been linked to greater efficiency of food breakdown. If we move across the spectrum here, we get to carnivores. Carnivores here are predatory animals, and so they have a type 2, largely predominantly type 2 fiber phenotype that allows them to generate rapid, powerful bite. And that's what they do in their ecologic context when they're preying after animals. This is pretty much what we know, however, about fiber phenotype in the muscles of mammals. I just put this ribbon here just to say type 2M, we know this is a unique isoform to jaw muscles, but we don't know that much about it yet. We know to some extent that it is faster contraction time than some of these down here, but it may in fact come at a higher tension cost than some of these over here. And the reason that we don't know for sure is because the studies have been done on different animals with different ways of getting at the question. So the question from my end is, if we think about the left-hand side of this schematic as a high-endurance, fatigue-resistant phenotype, right? During feeding, this is the primary selective pressure here is going to be high-endurance, fatigue resistance. And on the right here, it's power during jaw closing. That's the primary selective force here. question for me is, where do primates fit along the spectrum? Because we know virtually nothing about the answer to this question. Although I should have said that we know humans. We do know, have some good work on humans. So I went back to the sooty mangabey, that hard object feeder, as a naturalistic model because this sooty mangabey allowed us to test between two prevailing hypotheses about fiber phenotype in the jaw muscles. One is called the frequent recruitment hypothesis, and this is, in this case, these sooty mangabeys, they're processing these very large, hard saccoglottis seeds. It's a high-frequency behavior. They're doing it all day long. You might predict that they would have a slow fiber phenotype over a fast fiber phenotype because it's a routine behavior. And so you don't want those muscles to fatigue. Alternatively, high bite force hypothesis would predict that processing these seeds would require high occlusal forces, power, and so we might predict that they would have a type 2 phenotype that would be the predominant fiber phenotype to allow them to generate the power that they need to reach these hard seeds. And earlier this morning, somebody asked about food material properties. These are the seeds that I'm talking about. This is the shell casing. This is the internal architecture of this shell. It's very complex. And the material properties, the Young's modulus for this, is a little bit higher than prune pit and not quite as high as popcorn kernels. So they're about, sorry for that. They're about 200 megapascals compared to 180 or so, 160 or so for cherry pits, and 300 plus for popcorn kernels. So none of us in this room would want to put any of these things and try to break them because they would break us first. So, and again, they're doing this all day long. All right, so we got an adult sample of some sooty mangabeys. We had males and females, and we quantified fiber type distribution and the relative abundance. And we sampled regionally from temporalis as well as the masseter. So we did the superficial anterior and deep anterior and the superficial posterior and deep posterior as well as the anterior superficial masseter. These sampling sites we chose because we have EMG data on macaques, baboons in these regions. We did immunohistochemistry on formalin-fixed tissue. We got serial sections about 5 microns for each muscle block. We stained for 4 primary antibodies against those 4 myosin heavy chains, 1 beta, 1 alpha, type 2, and type 2M. We didn't differentiate across the type 2s. We just said type 2 and type 2M. And we counted and ranked fibers according to stain reaction intensity, either no reaction, weak, intermediate, or strong, and we measured fiber areas. So this is a sample from the sooty mangabeys. NOC is the antibody for 1 beta in the left-hand corner here, MYH6 the antibody for 1 alpha, 2F4 is the antibody for 2M, and MY32 is the antibody for type 2. And right off the bat, you can see here that MHC1 alpha, there's a lot of it. And I'm pointing that out to you because before this work, 1 alpha was never identified in a primate jaw muscle. It had been identified in humans and in non-human, some non-human mammals, but we didn't know whether we would find it in primate jaw muscles. We not only find it, we find it in abundance. Second thing I want to point out, again, these are just serial sections of the same fascicle, is that 1 beta and 1 alpha are staining with intermediate to dark intensity here. And when we look at the type 2, the faster fiber type, we see no staining. They counterstain. These fibers that stain here are counterstained. They are unstained here, and or lightly stained for type 2. The picture here is actually of a slow phenotype. This is the anterior temporalis. If I put up superficial masseter here, we wouldn't find hardly any type 2 at all. We found almost uniformly type 1 alpha and type 1 beta, 90%. So when we think about that hypotheses that we were differentiating between frequent recruitment and high occlusal force, we did not find data that would support the high occlusal force hypothesis for this hard object feeder. When we look at just a pie chart here, type 1 is predominant over type 2, and when we compare these sooty mangabeys to macaques and baboons, you can see that they have a slower overall fiber phenotype than either of these two species. And we can break the muscles down, and you can see that largely that slow fiber phenotype is being reflected in the superficial masseter. So they have a slow superficial masseter muscle. So what does that mean? Well, sooty mangabey jaw adductors, and I can say this now having looked across both regionally superficial anterior masseter, posterior masseter, different regions of the temporalis, they show a high prevalence of 1-alpha fibers. Remember, those are the fibers that we find almost 100% in the kangaroos that are grazers but not ruminants. So it does, we find them in rabbits as well and herbivores, but they're in humans as well. But these are fibers that provide a faster fiber population than 1-beta. They have a higher contraction velocity. They're slower than type 2, but they have greater endurance. They don't fatigue rapidly, and they come at a low tension cost, which is potentially beneficial for an animal that sits all day feeding on these seeds, nuts and seeds. They also show a high prevalence of hybrid fibers, and we know from the literature that hybrid fibers is more common in the jaw muscles, but I can tell you that while we have now looked at fiber type across 16 specimens, 4 species of Old World monkey, we've looked at apes, chimps, gorillas, and pygmy chimps. We've looked at New World monkeys. And the prevalence of, in fact, we are essentially not finding pure fiber types. We are finding only hybrid fibers. So this is a very interesting finding. We don't know what it means yet because we don't know enough yet about what these fiber type phenotypes mean. But the fact that we see this predominance of hybrid fibers suggests that they are playing some sort of a role in modulating the muscle function. And the question is why we see this such prevalence in jaw muscles and not in limb muscles. We also see regional variation. We do see differences between the temporalis and the masseter, and this does suggest some level of functional diversity between these two muscles that we don't currently have a good handle on yet. Is the masseter more slow? Is it because it stretches more in gape? Temporalis is a muscle that's recruited for vertical bite force. It's a good vertical bite force muscle. It's a faster muscle. We see that overall. These are questions. So where do we go from here? Well, we want to go back to the architecture data, and I want to say that all of the architecture data that I showed you are static. I got those data by collecting architecture on dead cadaveric tissues, and they allow us to make some estimates, and they allow us to put those estimates in models. But what we really want to know is how that architecture is changing dynamically throughout the gape cycle during feeding, and how those changes impact muscle force output and velocity. And when we're thinking about inputs into the model, this is the kind of input that we need. You'll be hearing at length about some really very cool data that Myra has been collecting on dynamic architecture. Is hard object feeding consistently associated with a slow jaw muscle fiber phenotype? So if you have large cross-sectional area and you're adding muscle mass, that's metabolically expensive. It would be economic to not add muscle mass and to rearrange the internal architecture, but we don't see that. So one question we have is, well, perhaps having a slow fiber phenotype offsets, counters the metabolic expense of adding muscle mass. Well, what about tufted capuchins? Those are hard object feeders. And to complicate life even further, we're not finding the pattern for tufted capuchins that we found for sooty mangabeys. In fact, we're finding that tufted capuchins have more of a fast fiber phenotype than the sooty mangabeys. This is just from Cebus apella, a tufted capuchin. We just got these data a couple of weeks ago. These are the same antibodies for 1 beta, 1 alpha. This is MY32 for type 2, and this is 2F4 for 2M. And you can see that we have a lot of 2M here, and it's picking up quite intermediate. This is fixed tissue. I have to say that we are probably underestimating the signal here. It's not fresh tissue, but we're getting beautifully dark stained cells here for MY32 and a lot of unstained cells for NOC and a lot of very lightly stained cells for MYH6, meaning not a lot of protein there, not a lot of signal in that. And lastly, what's the combined influence of fiber architecture and fiber phenotype on jaw muscle function and performance? So I said earlier that all of the architecture data that we have been assuming that cross-sectional area is comprised of fibers that are all of the same fiber types, but we now know that these muscles have different amounts of type 1 beta, different amounts of type 1 alpha, and so on. And so what we want to do is we want to combine that architecture with the fiber type data so that we can adjust the cross-sectional area to reflect the actual fiber types that are in the muscle. And we really want to start to understand why hybrid fibers are so prevalent in the chewing muscles and what their role is in fine-tuning muscle function. And with that, I just want to thank again the dental school, Piracicaba Dental School, University of Campinas, and the organizers for inviting me, and also all of my collaborators and the postdocs and students who have collected the bulk of this data. Thank you. Happy to answer questions.
Can I ask a question?
No, no.
Okay. Thanks, Andrea. So the hybrid phenotype muscles in the feeding system, do you think that enables them, that reflects plasticity or enables them to be plastic, and over what kind of time frame? So if an animal used its muscle differently, the fiber type would change, and how fast does that happen?
So that's a good question. I think that so first of all, we know muscle is very plastic. It's much more plastic than bone. Fiber types can be altered in a very short period of time under certain conditions. So for example, work by Williams and Goldspink where they fixed joints in certain positions. So they fixed them with one side of the muscles elongated and one side of the muscles shortened. And they came back in a week or a couple of weeks. And you see a shift in the percentage of different fiber types from simply casting in a joint in a position that elongates. So we know that they're plastic.
Sorry, and that was a limb muscle? Those
were limb muscles. We don't know the answer to that in jaw muscles. And remember, I haven't even talked that we have other myosins we haven't even looked like fetal and neonatal and so on. So we don't actually know we don't know the answer to that. An interesting experiment would be to start developmentally feed different foods, and if you could do it with inbred strains, if you really wanted to do a controlled experiment and see whether you get modifications in these in the expression of these different myosins, but we don't know the answer to that. I think that the different the myosin heavy chain composition diversity has to mean that we're seeing different differences along the length of the fiber in terms of their contraction velocity, for example, which means a finer modulation than a uniform type 2, right? But
so if you can change fiber type in limb muscles by changing the way they're used, the fact that there's a great diversity in jaw muscles, is it unreasonable to suggest that they're using their muscles in a greater diversity of ways in the feeding system than in the locomotor system?
I don't think it's unreasonable to suggest that. I think it's reasonable to suggest that. But then we have to look at the cases where we see uniformity, essentially a uniform expression of alpha cardiac fibers in kangaroos. I think the question is why. If we're going to say that we have a diversity of fiber types, we see these examples where we have 100% one fiber type. Why? We know, I mean, in the soleus, for example, it's almost 100% type 1 postural muscle in humans. It's a uniform, it's almost 100% type 1. But it's not the norm. First, there's many more myosin isoforms in the jaw muscle, so I think that's part of the reason why we're going to see greater diversity. I also suspect that we have underestimated the prevalence of hybrid fibers across the spectrum because, again, in primates, alpha cardiac had never been identified before. So in the work by Carlson, early work by Carlson and Miller and Farias on macaques, they didn't look for 1 alpha. And the antibodies we know for 1 beta can co-stain with 1 alpha. So a lot of what they found that was 1 beta was probably also some 1 alpha. So yeah, that's my answer.
Piggybacking off of Callum's question, is the diversity of fiber types in the jaw musculature a result of there are so many more muscles in the limbs that can sort of deal with those roles, right? A soleus could be a postural muscle, while gastrocnemius could all be like a propulsive muscle, right? Is that why you're maybe seeing that diversity or not?
We've had this conversation. We've talked about this for a number of different times. Why do we see this diversity in the jaw muscles and not in the limb muscles? So absolutely, and of course, we have to stop thinking just in terms of humans with 1 lower limb, right? When we're talking about non-humans, we're talking about quadrupeds, or I'm not going to go to lizards, but or fish. But so we have multiple joints, right? And so the loads and the ability to accommodate to different substrates is being picked up, let's say, by multiple joints. Yeah, we have 2 jaw joints, bilateral. Some have movable symphysis. Are the substrates that we take in terms of the food objects, set humans aside, are those any less complex than the substrates that animals locomote on? You're the locomotor expert. I can't answer that, but I think that we don't really, we're just starting to understand food material properties. We haven't even really begun to look at the geometry, which is the complexity also of the substrate when you ingest. So it's a good question. I mean, yes, I've said that before. I've said that that could be the reason, but I don't know that that's the reason. That's a speculation. Yeah.
Any other questions for Andrea?
No. Okay. Thank you very much.
Great. Thank you, Andrea.
Thank you, Andrea. Now pause to break through cough in 50 minutes. 15.
Hello? Hello, 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 hello. Like this.
Okay, let's get started. It's my pleasure to introduce Dr. Adam van Kasteren. Dr. van Kasteren is doing a lectureship at Washington University in St. Louis. He got his PhD in Manchester working on wood biomechanics in orang nests, and then he went to Kuwait to do a postdoc with Peter Lucas where he worked on grit, I believe, and that's what he's going to talk about. He was at Leipzig for a couple of years as well. So Adam van Kasteren is going to talk about what's grit got to do with it, and that's a song actually, and he's going to start by singing for us.
I will not sing, but so this is a question that I found myself kind of bogged down in at the moment, but enjoying being bogged down in it. And usually when I go home to speak to friends or anything, the first question is people say, why are you researching, why are you talking about grit? Specifically at a conference that is based around kind of craniofacial biomechanics. Well, it's known to be kind of incredibly damaging to teeth, right? It produces big scars, even at low forces when you drag them across enamel. So much deeper than kind of fight lifts and other abrasive materials. And if it's so damaging to enamel and it is wearing them away so much, it's likely that it is a kind of a selective pressure, selective force acting on the kind of micromorphology and arrangement of the enamel material. This work here was conducted by Peter Lucas back in kind of 2013. And this is the model that he came up with for how grit might be wearing away at enamel, right? And so you have your hardness of the particle that's going to be sliding against the material, and you have the hardness of the material and the toughness of the material. The hardness is defined as the resistance to deform under indentation, and the toughness is just the energy taken to propagate a crack through that material. And the angle of attack here, which is this critical angle of attack, is basically determined by the toughness of the material that's being worn away. So if you have a hard particle that is harder than the material that it's wearing into and has an angle of attack that is above a critical angle, then you will get instantaneous removal of material, right? Little chips coming off, maybe a ribbon coming off, and that's detrimental to teeth because you are losing material with just one interaction between a particle and the tooth surface there. What happens if you have a particle that has kind of a lesser hardness than the material and or a attack angle that is lower than than dictated to by the critical angle by the toughness? You get a kind of piling up around the material, right? So this may end up wearing enamel down eventually over many, many attempts. But at that single contact there, you haven't removed material, you've pushed it around. Like if I was to take my index finger and bash it into a kind of lukewarm butter dish, right? It would move material around rather than actual remove butter from the whole slab of butter. I know, it sounds ridiculous, right? But it's true. And so what happens if your teeth wear away, right? I know I'm talking to a lot of dentists in the room, so I suspect they know what happens if your teeth wear away. It can have a big effect on an animal's life, right? An organism's life. If you don't have teeth, it's hard to chew foods, it's hard to break them down, it's hard to digest them. It is seriously detrimental to your life, as I'm sure this old fella can tell you. But we have some actual kind of examples from the literature as well. These are shrews here, that if you take them in the wild, they live for about 1 year, roughly. And the old ones, when you find them dead, have incredibly worn down teeth, right? They are not surviving long because essentially their teeth are worn down. If you then take the same species of shrew and raise it in captivity under clean conditions, where it doesn't have to deal with harvesting all its food from the dirty ground, they can live for 2 or 3 breeding seasons and their teeth don't seem so worn down. You have kind of, in these ring-tailed opossums, where you have high wear in their teeth, you generally see low survival. And the next example is a bit anecdotal, but I quite like it, is kind of, people have suggested that old elephants lightly starve, because they have these 4 great big molars, right? And they wear down, they move in a position, they wear down, they move in a position. And people have suggested that once you get to the last stage of wearing down all your molars, you move to a more marshy environment where the food is softer and you can break it down a bit more, and eventually you just can't process as much food as you need. So the cause of wear is a real evolutionary driver to the dental adaptations against wear, right? Also, if you have an agent of wear, as grit has been proposed to be, that may be responsible for at least some of the kind of microwear signal that you get, right? And I've nicked this from Kami et al. I quite like it. They did a nice study where they had a mechanical chewing horse where it would chew and chew and chew, and they would add different diets to it. Right here is Lucerne, which has, very... Or is it alfalfa on this side of the Atlantic? I'm not sure. Alfalfa? I don't know. Anyways, it has very low kind of silica levels internally in it, and it's quite tough to eat, but it's usually thought of as an easy option for horses to eat. It produces kind of some striations across there. You add in grass, there's also striations, grass and some rice husks, which have more phytoliths or silica in it. You get more striations. This is where they added sand into the mix as well. Then you get a really rough, complex surface texture that has got more pits and more striations in it. And this is just the adding of sand. And the last one here that you see where there's kind of striations, they're all kind of in a similar pattern, but they're all coming off. It's just general attrition, kind of tooth-on-tooth wear, right, in what you have there. So you can go from this to this by just adding a bunch of sand to the diet, right? Sand is essentially a proxy for grit. And so what I'm interested in kind of thinking about is that you have a microwear like this. I just chose a few primates from Scott et al. Here and averaged up the kind of anisotropy, which is the striations going all in the same direction, and the complexity, which essentially is the more complex you have, a more rough and a more kind of bumpy surfaces with more pits in and this kind of stuff. And these two here are quite interesting because these are often considered our kind of hard object feeders, as we were talking about earlier on. We have the kind of robust capuchins out here and the sooty mangabey out there. So I want to know how much influence, if at all, the grit is having on the kind of microwear signal down here. And so today I'd like to break my talk into kind of three basic questions. How are teeth adapted to everyday threats? What kind of micromorphology have we seen in teeth? How we measure the mechanical properties at the correct scale to get a good answer to these questions. Then I want to ask what's doing the most damage to teeth. What is the likely ecological kind of damager to enamel surfaces, and what is the ecological prevalence of such threats? Because there's been some great work looking at where dust is in the environment and stuff like this, but I think we can go a little bit step further and look at it where it is on the actual foods and how it is going to be deposited within the mouth. Today, for any of the biologists out there, I'm going to be incredibly mammal-centric, right? I'm not going to talk about any reptiles or any kind of fish or anything like this, but there is great research on their teeth as well. And I'm going to stay very much in the kind of micro-nano realm. So I'm not going to talk about gross morphology or kind of macro wear or anything like this. It's going to be really small and really tiny, but fun at the same time. So teeth, right? I suspect all the dentists in the room know what teeth are. I've stolen this off Paul Constantino. Teeth are essentially migrated vertebrate exoskeletons that have come inside your mouth. Why have I put that up? It's interesting because the kind of evolutionary root of teeth is something that is outward facing, right? It is kind of designed to reflect the particles and the bits and bobs that will be bumping into it on the outside from the external environment coming in. They come in a vast kind of array of forms and morphologies, but the main thing is that the tissues that are involved in them, whilst they do change a bit, and I will argue that they can be kind of adaptively tweaked to the threats that are approaching them, the tissues remain remarkably similar, right? You have, dentin and enamel, basically, and enamel is that outside covering there that is going to be mostly chipped away at by threats to teeth. Oh, there you go. And then you have to know a little bit about the kind of nanostructure and the microstructure of teeth to really understand how we're going to get at this research. Again, apologies to dentists in the room. So here we have the kind of base unit of enamel, right, which is this hydroxyapatite crystal. And these crystals are lined up, thousands of them are all piled into kind of circular or semicircular horseshoe-like arrangements here called prisms, or rods. Sometimes they're called rods. Here they are in a real tooth. You can see them right there, the hardness taken. And on the outside here is this kind of 100 nanometer sheath, which is meant to be completely crystal-free, but recently there was some cool research showing that there's actually kind of crystal bridges that are built across these things, which may add an extra level of kind of toughness between them. But in general, this sheath is, no crystals are like really packed in there, and the crystals tend to face a little bit like this. What I want you to remember from this image is this something I have drawn very schematically, right? So everything looks very tidy. It's all kind of facing in the right direction. I've added in a couple of wiggles there to indicate that not everything is completely kind of organizationally perfect, but this is what it looks like in real life, right? When someone slices through one of those prisms, you get kind of organized chaos, right? You have bits going in one direction, you have bifurcation so that they aren't completely together, you have spaces between them so that between the crystals there will be kind of some proteins, a lot of water. So the idea is that here is not a perfect of arrangement, right? It is complex and it is a little bit crazy at the same time. Moving up a level from that kind of what you could call micro/nano to the actual like structural arrangement of these crystals, the way these are mostly arranged differently is you have a line right here. Back here towards the enamel-dentine junction, you have kind of decussated enamel, right? Which is enamel prisms. They follow kind of a wavy pattern, and they're slightly out of sync with their neighbor. This is thought to arrest cracks slightly, meaning there's more energy needed to direct the crack through this decussated enamel. When you get to the edge here, everything seems to straighten out. It's a united front against kind of wear particles. I often think of it as, if you're a history buff like me, a kind of shield wall against like invading armies, because here all the kind of prisms are facing in one direction so that you have a kind of united front. And this is thought to be better against kind of wear and the breakdown of tooth enamel in general. Now we have to talk about mechanical properties a little bit, because I am a big fan of biological composites because they are remarkably fun to look at mechanically. You have the base unit here is a hat crystal. It is very stiff at about 125 gigapascals. If you put thousands of them in certain arrangements and then take the bulk number for enamel in general, it drops by about half to about 60 gigapascals. The opposite is true for toughness, right? Here you have kind of a very low toughness of 5 joules per meter squared. You add in kind of structural complexity and the way that those cracks can be arrested as they move through the material, and you get a much higher toughness, circa 200 joules per meter square, which isn't amazingly tough, right? But considering this is essentially a ceramic-like material, it's pretty tough. It's pretty good at resisting wear. And threats to teeth, as well as mechanical properties, come in rather different shapes and sizes. Here we have very small kind of abrasive particles that are going to be kind of pounded into the enamel and then slid along it. You have much larger particles, and these are also hard particles. And where they hit the enamel, the stresses are going to be concentrated underneath. The fracture is likely to start at the enamel-dentine junction, because that is often a weak spot in humans and some primates. They have possibly tufts there, which actually are pre-cracks in the enamel, which are used to absorb the kind of strain put into the enamel and also direct the cracks into the decussated enamel, right? So you have strain absorbing some of that force, and then you are directing the crack into the material that is hardest to get a crack through. And then if you have a kind of soft material that might smother a crown, you might be getting the stresses building up in different areas of the tooth, further away from where the actual point of compression is, because the kind of dentin underneath is bowing out and the pushing pressure on the enamel further away from the point, so you get a crack that comes not directly underneath the particle, but further away. And this leads to a veritable like cornucopia of damages that can happen to teeth, right? You have, margin cracks and radial cracks and chipping, where hard forces come in and taken off like in a scallop-sized chip off the edge. But the one that is most common, that most animals and us and you know, lots of people have problems with, is abrasion, right? Because obviously you make thousands upon thousands on tooth on food, tooth on tooth, tooth on grit contacts throughout your chewing life, and they have thousands upon thousands of tiny particles within there, so that builds up to a decent amount of wear, and it can become a problem, as we showed with toothless shrews and old elephants dying and stuff like this. So this leads you to think, if you're going to look at abrasion, you have to look at the mechanical properties at that scale, right? We have to look at tiny measurements of mechanical properties, and abrasion is going to happen with, you know, micron, 2 micron sized particles, or maybe bigger, but 2 micron sized kind of striations cutting across the middle, usually the middle of a prism, right? If you have those prisms lined up and the only kind of the free bit of the prisms is just a small 100 nanometer border around it, most time you're dragging across the prisms, right? So you're not cutting in between prisms, you're pushing a particle over the surface of enamel and cutting through a prism. So we need to know the modulus and the toughness on that scale, the intra-prismatic scale, right? We don't need to know what the bulk enamel modulus is or the bulk enamel toughness for abrasion. We want to know intra-prismatic toughness. And so that's quite hard to get at because it's very small, right? Prisms are quite small, you know, 5 or 6 microns across. You can't just, well, you can use a ion beam to clean everything else off and then cut through it, but if you don't have an ion beam, it's very hard to do that. What we did have when we did this in Kuwait was an atomic force microscope, which are super cool machines, tiny little cantilever beams that you can then you can just bounce across a material, essentially, and then work out the viscoelastic properties at a very, 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 very small scale. So we did lots and lots of taps across a prism. But before we did that, we thought we would check that the measurements we got from the machine using these viscoelastic properties actually matched up to kind of measurements we got from bigger pieces of material, to make sure we weren't just using kind of a random number generator and getting the numbers that we wanted. So we did this. This is the on the y-axis here, you have the AFM modulus, which gives you estimates of the Young's modulus. I must say the Young's modulus, of course, is just the stiffness of a material, right? It's stiffness is the opposite of the compliance, so the higher the modulus, the stiffer it is, and the more elastic resistance it has. And then here we did independent modulus values, which were done with hemispherical probes. So we just stick that probe in to the material, you measure the force that you have when you stick it in, and then you just let it relax with that material, and you see how much the material relaxes around it, and you can work out a kind of instant modulus and an infinite modulus as the creep happens around that material. And what we saw was actually with these elastomers, these soft materials, it was all right. You know, that's a decent spread around the line, and then you move up to polymers here, these plastic polymers. It's when we actually really got up to these kind of stiffer, you know, the dentin and the enamel, that the numbers really came into alignment with each other, almost one-to-one. And then we were saying that for these stiff kind of like biological composites, this seems like a reasonable way to measure the viscoelastic properties on a very small scale. So that's exactly what we did. We tapped across a prism like this, and we got a lot of measurements. And then this is just the frequency of those measurements. And the first thing I noticed from this is what a vast scale of measurements, right? We have very, very kind of compliant areas down here and very, very stiff areas down here. Now, Spears, I think in 1997, predicted a range of moduli from computer FEA models of how he thought the crystals inside prisms were arranged. And he predicted a range that's not dissimilar from what we got from, and said that it was the orientation of those crystals in there. If there was more space in the way they are orientated, there'd be more water and proteins, there would be more room for movement, right? So that would reduce the stiffness of the material, because everything could kind of flow a little bit within it. We did notice that the highest frequency is really a great match for the kind of bulk modulus of enamel, which is really good. And these seem very high, but that might have just been a particular region where all the crystals are very tightly packed, right? It comes very high. It's very close to what you would see for kind of pure hap crystalline arrangements. This is interesting, and the reason I've put this up is because it shows there's a degree of deformation in there. There's a degree of movement between those crystals, which will reduce the stiffness, but will hopefully elevate the toughness, right? So then we have to come up with a way of measuring toughness with inside a prism, which means you have to create a fracture with inside a controlled fracture inside a prism. And this has been a problem because usually when people do fractures in enamel, it's on kind of indent scales. This is a micro indent here. And as you can see, the cracks come out from the edge of the indent, and it has almost traced perfectly around the edge of those prisms, right? So you're not really measuring intraprismatic toughness there. You're actually measuring the kind of toughness to skirt around the outside of the enamel prisms. And I put this up here because you can really see it in a grander scale, and even when they come off on the side parts here, I would argue they're skirting around the prisms. And that's just because, you know, a crack is like water. It will probably take the path of least resistance, right? So whatever you do, it's very hard to initiate a fracture that small. But Atkins and Patella et al. Have come up with some great models that are saying that actually when you cut a material, when you abrade a material, you do a sliding abrasive, it is part of the hardness, part of the angle of attack, but one part of the equation is the toughness. So if you know the angle of attack and you know the force that is both driving a rod into the enamel and scratching a rod across it, which you can do with a 2-axis load cell on a nano indenter, you can then work out the kind of intra-prismatic toughness, which is what we were going to get at. Oops. And that's exactly what we did, right? We made scratches under varying but constant loads, so 250 right the way through to 4,000 kind of micronewtons, and recorded both the force normal to the material and the force that was cutting through it, right? So we took a nano indenter, which has a defined kind of head to the indent, a kind of tip, and so we know the angle of attack. You drive it in a given force, and then you scratch it across. And you get lovely scratches. Oops, you get lovely scratches like this. And with some rather complicated maths, which I happily spare you, we worked out that the toughness within a prism came out at around 244 joules per meter squared, and the shear stress came at 1.48 gigapascals, which is pretty tough, right? It's pretty tough for inside a prism. But this mostly is just a kind of proof of practice that we could do it. Because what you can really do now is we can start to do a kind of comparative study across many mammals that eat many different things, hard object feeders, cows that eat grass, horses that eat hay, and lots of different things. And then we can start to think how their enamel itself has been adapted on a microscopic scale to their diet, which I think is maybe an untapped well of interesting kind of output. Just to show this, I thought I might go through a couple of interesting diametric opposite kind of diets to try and work out how we would predict or hypothesize the enamel might be adapted. If you're wanting answers from this slide, I can't give you them because this is something I would like to do in the future, but this is the kind of ideas that we have put together thinking about them. So a cow, right? It eats a lot of grass. Grass has both phytoliths. It has also covered in dirt. It is full of abrasive wear. They are really worried about those microscopic interactions with the tooth, and that is going to be what limits their tooth life. So they're going to want to have kind of adapted features on the intraprismatic scale, upping that toughness, upping that hardness, increasing the radial amount of the enamel. And that means that the selection is basically on the prism, right? It's not the way the prisms are right. It might be the size of the crystals in the prism, the way the crystals are arranged within the prism. Sea otters, however, live at sea, and they eat a lot of shellfish, and although you do occasionally see them with a very cute picture of them banging things on their chest for tool use, most of the time they're just cranking down with their mouths to open up, and they have much tougher enamel to do so. So they're worried about breaking their whole tooth, right? If they break their whole tooth, it's going to really limit getting that delicious crab. And they're worried about kind of the supraprismatic scale, right? They're worried about how the prisms are arranged in general, how that structure of the bulk enamel is going to absorb those big high forces and going to stop those cracks from pushing throughout out the tooth and causing whole-scale fracture. And therefore, I would argue their selection would be more on the microstructure than the selection of the internal structure of the prism itself. However, that's just a hypothesis, and hopefully someone will eventually fund me to carry on and do extra work on this. So if we can say that grit is scratching a lot, we've already talked about the sooty mangabey here. and how kind of it eats hard objects, and that's associated with complex textures. But within the model that I presented earlier on, nuts themselves are hard for sure, but they're not as hard as grit and kind of phytoliths and dirt and all those things. So what's doing the most damage to teeth? I'm going to ask, are nuts and seeds actually doing a huge amount of instantaneous damage, right? So when they instantly hit off the enamel, are they the particles that are causing the most wear? So to do this, again, I sat at a nano indentator for weeks, gently sawing seeds into very fine particles, and selecting through all these seeds until I could find one with a point, and then trying to mount it on a tip like this, which is, about 1 millimeter in diameter. You've got to get just the right amount of glue, because if there's too much glue, it's going to go wrong. You've got to have it in the right orientation. It essentially means that you spend weeks and weeks getting it on there, look at it under a microscope, and decide no, and then you start again. You do it again and again and again, but eventually you get something nice. This is actually a piece of Saccharomyces seed, which is on the outside there. Let me just tell you, this is actually a point, so this doesn't matter. It's like a pyramid, a big pyramid. You mount the seed. You then take an enamel surface, which is polished to a very, very fine scale, so you've got it as flat as you can possibly make it. We make what's called a landing strip on it, where you put indents at 20 microns along there, and then you do 80 microns, and you do 20 microns like this. So you literally have a landing strip like an aeroplane going in, and then you just simply take that and you scratch it across there at varying forces. The reason we put a kind of landing strip is so we know where to look for the scratches afterwards, right? Because it's a big tooth surface area. These scratches are about 16 microns long. You need a bit of indication of where to do it. So you scratch the enamel. You take the tip before and afterwards, and you do EDS on it, electron diffraction scatter, and you can work out. We've seen one this afternoon. We went to someone's laboratory where you have this piece of kit. They're awesome. You can do a map all the way across the image, and it tells you the kind of elementary composition across it. We did it before and after to see if there was any kind of calcium-based particles there, because that will tell us if little bits of chip are breaking off. And then we scanned the landing strip afterwards with the AFM, so we can see any very fine striations, any larger holes or pits or anything like that, to see what was actually in that area where we had been scratching. And in general, we didn't see a huge amount. We saw some marks, which is to be expected. Here's one. If I'm correct, this is the Saccharomyces mark, but that's not deep, right? That is 150 nanometers at its tip, right? That's not a deep scratch. It's not a wide scratch. It has what could be some pile up here. What's more interesting to me here is this is a Mesedia seed. Some work that Peter Lucas has done on his life is spent a lot of time working out how orangutans crack open those seeds. It's using their germination mechanisms and stuff like this. We scratched that against the orangutan enamel here, and we found this buildup like this, which is very strange, right? We were expecting it to be indented, not built up. And so we thought maybe that's seed, and we simply did that viscoelastic tapping of the AFM again across it, and what we got was, yes, we got the peak that showed us we had enamel there, right, at 65, 75 gigapascals. What we got here now is this extra peak at 11 gigapascals, and we said 11 gigapascals happens to be the stiffness of the seed shells that were being scratched across there. So what that means here is you have not a huge amount of wear in the enamel, but actually kind of a buildup where the tooth has been smeared across the seed has been smeared across the tooth. So you've got a buildup of that material there, which is kind of interesting. It'd be interesting to know how the area underneath has been affected, but rather than having the nuts and seeds really wear away at the teeth, it was almost the opposite in some cases. We did, however, and this really was interesting, find enamel chips on the outside of the teeth. I think we found these on 2 of the seeds being scratched across. They were very small. I mean, this is a micron size here. They're small. They're usually chip-shaped, unsurprisingly. I don't have a mechanical model to explain why this is. We just think that, when you get down to a small enough scale, everything that appears smooth is rough because you've changed the scale. And they may be embedding in the seed, or the seed may be catching imperfections on the enamel surface, and you get little enamel specks like this. There weren't a huge amount of them, but they were there. That's interesting. It makes it more complicated. You are seeing wear, but you're not seeing a huge amount of wear. It's And so the basic conclusion that we had was essentially possibly the actual seed and cell material itself isn't actually kind of isn't making the big pits that we might see on hard object feeders at all. There was a challenge, though, which came shortly after, alas, here we go, is unpublished work. I'm trying to get it published right now. Then this came out whilst I was in Tanzania in the field, which shocked me quite a lot, but it was interesting all the same. It was by Shah et al. And they essentially rubbed aluminium balls across smoothed enamel surfaces and found that you got these striations, and within those striations you got very tiny enamel chips, like the enamel chips we got, which is awesome. But you got these very tiny enamel chips, and the authors said, look, they are wearing away enamel instantaneously, and therefore softer particles can wear away enamel, therefore grit isn't such a big problem that Lucas et al. And Van Kasteren et al. Suggest. However, we have come back. This is, this back and forth kind of tug of war science that we're into at the moment. But we've come back and said all that glitters is not aluminium. Essentially, once you take aluminium out of its non-oxygen atmosphere and you expose it to air, it becomes aluminium oxide as it binds, as oxygen binds to it. Aluminium oxide is a lot harder than aluminium, 3 or 4 or higher sometimes in hardness. And so, again, we use that magic EDS there, and we take this is a SEM of the surface. Note it's not smooth, it's prickly, it's got crispy bits that are like a kind of cooked chicken skin on the outside. And when you do an EDS on the outside of this crisp here, you get a little oxygen peak right there. If you go deep down in between these cracks here, there's no oxygen, there's just AI, and then you have an oxygen peak on the surface here as well. That to us indicates that there's oxide within that aluminium. If you look at the original Shah paper, they did EDS. They also have an oxygen present in there. And we did some nano indentation on this. And you see such features like this here, which is called a pop-in crack. It's a brittle moment where you're pushing, and then pop, it pops in. You get a quicker change in kind of displacement, then you get force. It's a brittle snap. That is usually indicative of a hard, brittle skin on the outside of the ball. And actually, we did looked at the average pressures as they grow, as you indent, and at like 5 nanometers, we are above the pressure that should be equal the hardness of enamel. In full disclosure, last week, just before I came here, these guys have also written back and said that we've messed up our maths here, which I don't think is true, but equally, they still say there is aluminium oxide out there. So if we've done the indentations wrong, we've done the indentations wrong, but it is still aluminium oxide, which is much harder than aluminium. That happened to me. I just pressed the play button instead of... So you may all be thinking, this sounds very, 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 very pedantic and not very interesting, but I assure you it has some kind of point. The clickers, there you go. Essentially, it's because we have in kind of anthropology or biological anthropology, we have this kind of mismatch, right? This disagreement between the microwave signal and the kind of morphological signals that you might get in these robust australopiths here. And what we are trying to insinuate is that maybe an absence of a hard object signal doesn't necessarily mean an absence of kind of hard plant material, because hard plant material might not be causing these great big pits. It may be more to do with kind of the composition of the abrasive particles that are in there. So if you have a large amount of grit particles in there that are also large in diameter themselves, they may be chipping off huge parts of dental enamel. But then if you have either more phytoliths or much smaller dust particles, you will end up with kind of striations. So there is still a dietary signal in here. It just might be less direct than to do with the mechanical properties of the actual food themselves. We suggested in that paper that maybe this would be, and this is super schematic, I admit, but it's just a kind of first principles idea. We have stolen the idea of Callum Ross that chewing is chewing is chewing, and you have the same chew cycle, and you have hard particles like grit on the outside, and just essentially it's more to do with the kind of shape and form of the food. If you have a flat plate, these particles are going to get embedded and then dragged across the tooth as you shear that material into smaller particles. If you have a more isodimeric shape or rounded shape or even an oblong shape or whatever, a large kind of bulk shape, it may be rolling across the tooth, which may be indenting grit particles in kind of different areas and producing more of a complex texture rather than a striated texture. Just an idea, something again that needs to be tested. We did consider that possibly grass seeds might be something that is being eaten by hominins because you have the high isotopic, well, the C4 isotopic signature from their teeth, which suggests that they are eating a large grass-based signal. People have said that this could be grass leaves, but actually seeds are also a good model for this, which has been suggested before by Dom Jolly and all these people, but we are just adding again, not Dom Jolly, he's a British comedian. So, didn't mean him. He might think it, but he certainly hasn't told me about it. Anyways, by Jolly back in the 70s, that he suggested, kind of small seeds might be useful. Here we have kind of building up. To crack a seed initially is very low force, right? But we don't want to crack, right? We want to grind. We want to get out the nutrients that's within inside that carapace. So you want to apply more force, do this kind of lateral movement that's going to start grinding. You also want as many seeds as you can get on there if they're tiny, right? Get as much in there, grind as much in there, more energy goes down your throat. And so we show that actually if you increase the amounts of seeds, if you get to like 32 seeds, you get a much higher force to the initial crack, and then if you want to be grinding, you're going to have to be providing a huge amount of force. Also, just here, this is just a single seed, and then you crack it, then you add more seeds to crack, and you get up the higher force. What's interesting is when you CT scan some seeds, not all of them, there are fight lifts all around the outside. Like, does anybody watch Doctor Who? This is kind of like a Dalek's outer surface. It has like bulbous parts of it that are full of silica bodies on the outside, arming the outside. So what actually is happening that might cause those striations is that when you're crushing a seed across a flat plane and it's being pushed out from the centroid, you're getting striations and you're grinding that around. So these might be a good idea. Again, just a suggestion as to how we can start to bridge that kind of mismatch between the microwear and the morphology that is there. So what damage do these seeds do? In answer to that kind of second question, nuts and seeds, if you believe our scratching test is not that much, plant-derived stuff, if we look at the model from the Lucas work, this will mark enamel for sure, and will kind of change rearrange the enamel, which may make it weaker for future processes to start wearing away at it. But instantaneously, it's not doing a huge amount of damage. Again, if you have enamel, which is also going to be in your mouth, when your teeth are colliding together, you're going to make chips of enamel that are going to be swilling around in your saliva, get mixed in with your food, and then those tooth-on-tooth contacts are going to cause you to have striations made from this. Again, they just seem to be thin kind of grooves, and they're not chipping away huge amounts. So the real kind of bad boy of abrasive wear is this geologically derived stuff, right? The quartz that's in the grit, maybe field spars. You got kaolinite. We just don't know the kind of properties of these small particles. We also don't know a huge amount about the prevalence that they are in the environment. We have ideas and kind of long-held beliefs, but I don't think there's a huge amount of data out there. It is a hot topic, though, what damage this stuff is doing. You have, the Kame stuff that I showed you already. You have our stuff that's talking about grit and fighter lifts. And you have, Thomas Kaiser stepping in there, Peter Lucas, and this one recently. This one is what I call a bottle of wine paper, because I read it and had a bottle of wine immediately, because I was really like, oh my goodness, my last 2 years have been useless. But it's an interesting paper. I don't completely agree with the title, but that's what science is. And so I wanted to look more at what the actual ecological prevalence was in there. I want to get out and measure some work. There is some really good stuff by Peter Ungar, putting up dust traps in different canopies, in different seasons, in different locations around the world. And he showed that actually there was a decent amount of particles that are being drifted around in the air around us, right? And they were landing on sellotapes and they were being collected. Great stuff. But I don't think plants are an inactive agent in their existence, right? Plants have, sticky surfaces. They have, silicous hairs all over them. They attract dust. They repel dust. They have, waxy cuticles because they want to keep their solar panels clean, essentially. Different parts of plants will attract different parts of dust, will trap things differently. So I went out to Africa and decided to look at chimpanzees, which don't seem like the original model for looking at dust. We did some other work on them, but actually it was these two environments particularly that I was very interested in. Chimpanzees spend a huge amount of time what I would call gleaning dust off things, right? So not gleaning dust, sorry, gleaning fruit off things. They do occasionally dig around in the ground. They do some eating of termites and stuff, but they are ripe fruit specialists. They eat, the vast majority of their diet is made from fruit that they've got from trees, and then a huge amount after that is leaves that they have from trees, right? So we're looking at wind-deposited dust, wind-deposited grit. It's all over the place. And so it seemed like an interesting set of environments. And Gogo here houses the biggest population of chimpanzees in the world. It is in this lush Kibale Park. Admittedly, Kibale National Park is a little bit small, so expect some edge effects from kind of camps and stuff around it. But in general, it's wet, it's dank, it's chimpanzee heaven, right? It's full of fruit. All year round, they can eat luscious fruit. They fall back on big peach-like figs, which are the poor things that they have to eat, these delicious fruit. And then you have more like chimpanzee hell here, which is Ugala, which is out in Tanzania. This is either the first or second driest site, depending on which primatologist you talk to. It has incredible seasonality. I got there in May, and it did not rain until October, and everything set on fire and went. And so there was a huge amount of fire. There's a lot of dust in the air. There's a lot of wind. There's a lot of open place. It is a kind of mosaic habitat. You have by the rivers, you have these thick forest patches, but out here, this is really open field, kind of open forest scenario. The hypothesis being that here is wet and closed, not very dirty. Here is dry and dusty and windy. It will have more grit on it. This is just to show that the two populations here are eating foods of different mechanical properties, especially when you look at the exocarp. That's the kind of peel on the outside, or the mesocarp, the pulp, and the leaf tissue, and the mid-veins are moderately cinema. There's a bit of significance there, but I wouldn't build too much biological significance into it. But it's this here. It's that outer kind of husk or peel is much tougher for these savanna chimpanzees. The same thing with the stiffness. It's much, much more stiffness. You're almost in the woody realm right there compared to the mesocarp, which is different. They also eat in different areas, right? So the isotopic signatures are different here. The Isa chimpanzees are eating in more open habitat. In the dry season, they come out from these kind of forest, ravine forests, and they come out and pick fruit from the savanna because you actually have an increase in fruit in the savanna environment during this dry season. So arguably, they're getting more from the savanna, which is where we would expect to see more and more grit being built up on the outside of the plants. How do you clean a plant? That's a big question. I talked to an uncle of mine at a wedding. I'm Irish, so we were both a bit tottery at the time, but he told me you can't just wash plants, silly. You have to really clean them very well, which actually means you have to put some kind of adhesive layer on them and then peel it off, taking everything out with it. That led me to some research that came out of Chernobyl, which is a big explosion in Eastern Europe, and a huge amount of radioactive waste was spread all over Western Europe, and so people wanted to know how much was being absorbed by the plants. And if you want to know how much heavy metals are being absorbed by the plants, you have to make sure there's absolutely none on the outside of the plant. So you have this plastic cleaning method where you make up this liquid plastic. It's very easy to do. You just add ethanol. You go around those two different habitats, climb up the trees to the point where you would find the fruit. You paint, literally with a paintbrush, the outside of the fruit, and you let it dry. And then you can simply peel it off the fruit, and you have a little time capsule of particulates. In my head, it was going to look like this. In real life, it was much harder because you have mites and worms and, all the different horrible, like, dandruff, skin, the lot that's been there. But let me just show you a SEM picture of this is a plant before cleaning. You see all that, like, gubbins on the outside, and then you clean it right there, and you just have a really clean section of plant. So you can do this. You take it back to the lab. You can simply dissolve that plastic in ethanol. You get this kind of mixture of all the horrible biological and mineral components that you want there. And then you can simply either... Whoa. Well, you've seen the punchline. You can simply... You can get rid of the biological component by either burning it off with acid if you have a very small sample, or burning it off with heat if you have a large sample. And then you can use these heavy liquids, liquids of different kind of densities to float off the phytoliths and stuff like this, so you can have a pure mineral content at the bottom. And this is all stuff that's floating around in the air and is naturally landing on the thing. And then you take it back and you analyze it. I use something called the rapid phytolith counting method because I don't want to count, several million particles. You basically take a subsample from that sample, you plonk it in there, you count how many there are per area, and you can work out a particles per millimeter, which is exactly what we did. This is the kind of sample you get off the outside of a Celtis milbradii leaf. All these big kind of see-through bits here are quartz. Then you have biological derived stuff like phytoliths, and you have some kind of like diatoms and stuff like this. And then you have clays are meant to be in there, but they disappeared somewhere. There you go, clays. These are things like kaolinite and stuff like this. I can't speak for how these are wear abrasives because we will only have kind of bulk samples. I don't know how they're going to work on a particle scale. They could be hard. They could probably be soft, though, at the same time. And this is what we saw, and it was rather different to what I was expecting. In green here, you have Ngogo, and in yellow, you have ISA. And then when you look at particles per millimeter of just everything in general, Ngogo significantly has more particles per millimeter than ISA, right? Which means the wet rainforest where everything's clean all the time is actually a wet rainforest where everything's dirty all the time. And the kind of open habitats here are consisting of less particles than you would imagine, than I would have thought. The reason for this is I think wet things probably attract more dirt, right? And the rainforest is a layered, kind of complex scenario where water comes in from the top and gets washed down and collects all the dirt, and then it sticks to the outsides of foods. When you break it down into rough categories like leaf or fruit, you tend to see that fruit have generally more particles, sometimes significant, sometimes not, than the leaf. Makes sense as well. Leaves are solar panels. You don't want tiny little reflective bits on the outside bouncing light off in all different directions. When I thought maybe they were different sizes, maybe the ISA particles were just much bigger, and therefore we were seeing less of them. Actually, when you look at the diameter of ISA, and these are a lot of particles we analyzed here, you tend to see that the mean and the median isn't too far different of these particles. It's small. It's very small. That's 5 microns. This is stuff that's being blown around by the wind, right? This is what they call aeolian deposited dust. So here, dust is basically everywhere. It's on all those plants and all those leaves and all this kind of stuff. I know I have spent a career telling people the blooming obvious, but it's all kind of morphological similar. It's you're not seeing huge differences in the size difference between those open and those closed habitats. I say open and closed habitats. I've looked at 2, so let's not get carried away, Adam. It could be very different in very different areas, but I'm just overemphasizing the data. I think the main difference might be between kind of gleaning, like these chimpanzees are doing, taking food out of there, and extractive foraging. And that's what we started to look at in Brazil with the capuchins, right? The kind of robust capuchins. They are often called seasonal hard object feeders. They fall back on these really hard objects. And they often exhibit a classic kind of hard object feeding signal, up with the complexity and down with the anisotropy. But we were kind of seeing behaviorally that actually some of the objects they were taking weren't particularly dealt with hard objects. They were tough objects. They had more like toughness involved in them and stuff like this. So what might be adding to this hard object signal that is causing it? And I am going to argue that in some cases they are eating very dirty foods, and the dirty foods might be adding to this kind of complex signature. Now, in order for this to work, we need all the robust capuchin species to be eating dirty foods, right? And they eat very different things in the different environments. We spent some time looking at Atlantic forests at Sapodgeus nigritas, and those guys were eating mostly in the canopy, so we didn't suspect them to be eating hugely dirty foods. You know, this is aeolian deposited food. Food shouldn't have large morphologically large kind of dust particles. Oops. And then we looked in the kind of the mixed cerrado habitat here with Sapodgeus libanosus, and then we went up to Suriname where we looked at Sapodgeus apella, and they again are in the rainforest environment. So if grit is adding to the complexity, we would have to see it regularly in the diet for long periods of time in the diet and across all these different ecological biomes. So looking at the ecotone of the Cerrado and the Caatinga, we have here looking at a sample I took from an anvil. The anvil is just a place where they are breaking the nuts. These guys are the tool users that are cracking open those palm nuts. that's some decent grit that's on the outside there, which is unsurprising because most of these things are kind of two rocks smashed together. I just want to point out this guy here. This is 200 microns scale bar, which is very small on this massive screen. This is a huge piece of grit, right? That's like, 400 microns in diameter. They're big pieces. There are also tiny pieces in there, but there are great big bits of quartz. We know these are quartz because this is cross-polarized light. Anything that's gleaming white is quartz, essentially. You look at the dust from a tuber. These guys love to dig up the tubers from the ground, bring them out. They clean them. They strip them. This is from a tuber that has been kind of stripped. There's still a lot of dirt on the outside there. There's a huge amount of grit. There's a huge amount of quartz. And they are large particles again. Again, that's 200 microns at the top there. And this is just dust from grass, right? The capuchins like to pull up grass and then eat the kind of fresh new ends at the bottom there. Well, that collects dust, and it's like a little dust trap in itself, and you get large particles like this. And this is a much smaller scale. This is 50 microns right here, and this is kind of dust from Marmelade de Bois, and that is more to the scale of what we were seeing in Africa, right? That's aeolian deposited dust, and unsurprisingly, these came from tree parts as well. Very quickly, how do I know it's dust? Well, you have a spectrum of pure quartz here. This is FTIR. You basically fire an infrared beam at a ground-up sample, and you get a spectrum back. If you see something like this, which is a double-peak quartz signal here, it's called the quartz doublet. And then you get this peak here. You think, I've probably got quartz in that sample. And if you look from brushed nuts, which is just simply where I've brushed the outside of a nut that they've been eating, you get, look at that quartz doublet there. There's also some other stuff in there, but that's also a peak for quartz. So I would say, yes, quartz. You have here a brushed tuber. Same thing again. You get that quartz doublet, that peak. And right here you have a brushed insect stick, which is just a way of saying a stick that they've been biting into to get insects out of. And this isn't one that's dropped to the floor and I've picked up. This is one I've climbed up and taken right next to where they were eating. So this is kind of the bark itself maybe acting as a bit of a dust trap. None of this should come as a surprise because this is kind of, they are extractively foraging from dirty things in a very sandy environment. But then what about Atlantic forest, right? We were looking at Nigritas here, and we were having a bit of problem with some habituation of the monkeys, but they were spending a lot of time in the dry season when we were there falling back on bromeliads and palm fronds, right? So essentially bromeliads are little ecosystems that are up there. They're collecting a whole amount of dirt, a whole amount of water, and so I could take that, clean the sludge off, clean the water out, burn it down, and see what was in there. And again, we cleaned the palm fronds with plastic, and this is what we got. Again, this starry night of kind of quartz, right? That's a huge amount of quartz in there. And look at the size difference. There are very small things in here. Again, this is 200 microns. That isn't as big as we were seeing earlier on, but that's still pretty big. It's not going to be the kind of stuff you get kicked up during a windstorm. Again, it comes out as looking pretty quartzy. There is some clays in there, which has masked this signal a little bit. This is just the water. So even if you were just drinking out of a bromeliad, you're probably ingesting a decent amount of quartz that's in there. Not so much on the valley pines. I would argue that this, again, I didn't clean this sample correctly because I am seeing a quartz doublet here, but there's also a lot of phytoliths in there because if the phytoliths, that rounds off that peak, so it becomes, because it's amorphous silica, it has a slightly different signal. I would argue that this sample is both quartz and phytoliths put together. And then Suriname, we did this year, so I have not done any of the analysis yet, but I will eat my hat if these aren't dirty. Look, all these brown bits here are just mud, essentially, in the bottom. We collected the water and stuff like this, and they spend a lot of time during the dry season here eating these kind of like long-standed orchid bulbs there, which will be also kind of like dust traps that are collecting stuff. So the argument, oh, no. Let me just try and finish this off. Yeah. The argument that I want to make is that actually across all these these ecological biomes, we are seeing kind of either the presence of dust or the high presence of dust in kind of fallback foods. Fallback foods can be sometimes more mechanically challenging the process, meaning they're being chomped down on harder, and we're getting these large pieces of dust, these large pieces of grit in there, which may be increasing the complexity on the signal, even if you're not particularly eating hard foods. But to be confirmed or finished at some point in the future. And just to say at the end here, thank you very much for the organizers for inviting me. It's been great fun. I've enjoyed talking to you. I hope I haven't bored the socks off you. And I have to thank everybody who's funded me and let me come and work at their field sites and stuff like this. And I hope you've enjoyed it. Thank you very much. Oh, and questions. Nope. Yeah.
So, it was a great talk. I really enjoyed it. How important do you think, if at all, when you're doing those scratch tests, how important do you think it is when the animal is biting? So they put it in their mouth, they bite, you get some failure. Yeah. That large item is now a bunch of small pieces. Now, how important do you think the fact that they now have all of these smaller particles, that they're going to break down even smaller particles, and they're hitting at different angles and different parts, how important do you think that is? End of question.
I think it's very important, right? And what we were trying to get down to the lowest kind of principles in there. They were as small as I could make those particles without making them actually kind of microscopic. So, you know, what we're generally getting there is particles of seed that are kind of 200 microns across. We can't control for angle in that nut-based project unless we get some interesting lasers, but they are above our pay grade. So, yeah, I think for sure the different angles of attack is definitely a question, right? That is because that is going to influence how deep your groove is, whether it's going to remove stuff instantly or whether it's going to build up particles. If you have a larger item, according to current thinking, is that you're spreading the stress out too much to cause microwear, right? So it's only those small particles that are going to be interested, and that's as small as we could get to them. If you get very, very, very small, your particle is probably going to flow more than fracture. So if you get below about kind of, I can't remember what it was, maybe a 0.5 of a micron, you're not going to get a fracture. It's going to squish and it's going to like flow like that. So then it won't be a problem. But so I would say, yes, below, you know, a couple hundred microns to small, those small particles are going to be a problem. But I don't know if they will change the mechanics of what's happening there, because the actual point of contact between that particle and the enamel is still very small, right? It's still going to be a point off. So I can't answer your question clearly, but we've tried to get as small as we possibly can.
Thank you. Thank you. Good talk, Adam. Thank you. So you pointed out that sometimes the enamel isn't actually removed. It's ploughed up into a pile at one end of the groove. That's the first power stroke. Yeah. And then, so what happens when you take another tooth and rub it across the top of that? Seems like it's dying to be rubbed off. There's another microphone there, because I have another question, actually.
Good. Absolutely. Yes. So it's making it not a flat surface anymore, and it is dying to be rubbed off. But that is, if you're thinking about this as like an evolutionary question, and you can look at bulk kind of wear studies like that Carme study, sure, those fight lifts are causing that. They're kind of building up stuff and it's being sheared off, and you're losing tooth kind of material over a longer period of time. But it's about half, right? It's about half the time that in their experiments that you see when they add grit. It's about half the amount that they remove in a given amount of strokes. Do you understand?
So, yeah, no, I get you. So what you're arguing is that if you take a piece of grit and you do one power stroke, for example, you'll wear off some enamel.
Yeah, and that's
whereas
gone.
With the other one, with the phytolith, you'll do two power strokes, and you'll wear off the enamel.
Yeah.
Or
you wear half that build up there, and the other half is more plastically deformed further around the tooth. It's not an and/or, right? It can be a complicated process, which is begging to be researched, right? But that's some complicated mechanics that I don't know. So I would argue, yes, it is a indirect wear process, right? I don't think anybody's arguing that it isn't, but it's taking longer than grit. And if you're a tooth and you have natural pressures being selected on your enamel, you're going to worry about the stuff that's going to wear away instantaneous rather than a longer kind of, if it's prevalent in the environment. If it's not prevalent in the environment and something that happens once in a blue moon, not a problem. I can see I absolutely haven't convinced you of that.
No, not at all. Animals chew a lot.
Yeah.
And they chew thousands of times a day, and I wonder whether natural selection would see the difference.
But so, I mean, in those horse studies, they're chewing, 40,000 times, right? and you're getting half the amount of damage without grit than you are getting with the grit. To me, that's something that's a selection pressure that's acting, right?
Yeah, sure.
So, yes, I agree. I think fight lifts and stuff are a wear agent. And if they are twice as prevalent in the environment, then they are probably dictating tooth form. But if they're not twice as prevalent in the environment, or grit is as prevalent in the environment, that might be the natural selective agent there.
Gotcha. All right, thank you. So my other question is about bruxism. And
Right.
in dentistry, bruxism is a big problem, right? And the teeth wear down a lot. I have a colleague who I see regularly, and his incisors are almost completely worn down to nubbins.
I have a stress response in my teeth. I allow my two. If you see me doing this, I'm stressed.
When I'm talking to you. So when I ask you questions, that's what you should do. So bruxism, there isn't people, if they do it in their sleep, then there's not grit unless they're putting sand in their mouths before they go to sleep.
Yeah, but again, it's the same kind of process. If you have tiny bits of enamel, no two contacts. If I stand at this table and rub it like this, eventually I'm going to wear away a hole in this table.
Right. So
if I get a pickaxe and drag it across here, I'm going to get there
so
much quicker.
Your argument about bruxism is that the wear is due to not just the teeth rubbing against each other, rubbing sensu stricto, but little chips of enamel coming off.
Little chips of enamel, little enamel. The enamel isn't as flat as we make out in these experiments because it will have, when you look at a complex surface texture, right, you have great big peaks and deep valleys. They themselves are kind of like wear particles that are just attached to the rest of the enamel. Enamel on enamel is going to mutually deform, and that is going to cause change to the surface. It's going to rearrange the material, but it isn't probably going to instantly remove the material. Sure, sometimes if you have a long enough point, it puts it in bending, that's going to snap it off like that. But I wouldn't argue that. I don't think, otherwise people who did bruxism wouldn't have any teeth in like 3 years because they'd be rubbing it together all the time.
Okay, thank you.
No problem.
Any other questions for Adam? All right, thank you, Adam.
Okay. Okay. So, thank you, Anna, for coming. Here's the
thank you very much.
Okay, thank you. Good lecture. Good having you.
Yes, thank you for having me.
Berta is the next. Esse aqui, aqui, ó. Acho que sim. Tem que falar desse negócio aqui? Esse aqui? Não, é nesse. É nesse. Bom, eu vou apresentar. Tá bom. Acho que é do contrário. Só tem BGA, só. BGA não vai passar na transmissão. Não.
So I would like to introduce the Professor Roberto Camoto from Unesp, Faculty of Dentistry, Piracicaba. Roberta will talk about the morphophysiological behavior of alveolar bone repair in rats. Okay? Roberta, thank you for coming.
Thank you very much.
Okay.
Okay, thank you. Thank you. Good afternoon. First of all, I'd like to thank you very much for the organizing committee for the opportunity to be here. It's a great pleasure and honor for me to be here trying to show some results of our lab. But now, my English finishes now. So, yes, because my English is very bad. So, now, I'm going to talk in Portuguese. I apologize, but please, yes, please, be my guest. Yes, so anything that you want to ask from me, you just ask, and I'll try to talk to you. That's because my English is very bad, so I apologize. Okay? But my lecture is Morphophysiological Behavior of Alveolar Bone Repair in Rats. My name is Roberta, I'm from Dental School, Aracatuba. O título, então, é o comportamento morfofisiológico do reparo ósseo alveolar em ratos. Né? Eu sou a Roberta, da Faculdade de Odontologia de Aracatuba. Eu dividi essa apresentação em 3 objetivos. Né? Então, eu vou primeiro falar um pouquinho da dinâmica óssea durante o processo de reparo alveolar, em condições normais, e também em presença de interferências sistêmicas. Porque, na verdade, o foco nosso no laboratório é buscar como é que é o comportamento do reparo ósseo em situações em que você tem um comprometimento sistêmico. Systemic interferences, like osteoporosis, diabetes, something like this. E depois eu vou mostrar alguns resultados do nosso laboratório. Como eu falei antes, né, para os pesquisadores, né, que estão aqui, I am an outsider. Eu sou uma assim, forasteira aqui, porque eu vou estar mostrando para vocês resultados em ratos, que são as coisas que a gente faz no laboratório, e falando especificamente em reparo ósseo. Tá? Eu gosto sempre, quando eu vou falar um pouquinho de tecido ósseo, começar falando da lei de Wolff, porque eu acho que é fundamental a gente não perder do nosso foco a dinâmica do tecido ósseo. Né? Então, o tecido ósseo ele é altamente responsivo a quaisquer tipos de estímulos que a gente aplica, como leis matemáticas, né? Como fala o enunciado da lei de Wolff. Então, acho que a gente tem que pensar a respeito das características do tecido ósseo, pensar a respeito dessa dinâmica do tecido ósseo e como ele responde bem aos estímulos que são aplicados. Então, a gente precisa tomar muito cuidado com relação a como é que a gente faz os procedimentos, especificamente no nosso caso, os procedimentos odontológicos, né? Em que você faz uma exodontia ou você faz uma instalação de implantes. Particularmente aqui são esses os modelos que eu vou estar mostrando para vocês. Lembrando que o tecido ósseo é um tecido conjuntivo mineralizado, constituído por matriz extracelular e também por células. Essa matriz ela vai ser composta, então, por uma parte inorgânica, que é a maior parte dela, em que a gente tem cristais de hidroxiapatita, e a gente tem uma parte orgânica, em que a gente tem uma série de proteínas da matriz, grande parte delas que vão ser o colágeno, e depois proteínas não colágenas que vão ser importantes para que a gente consiga ter a precipitação de minerais sobre essa matriz de colágeno. Eu costumo brincar com os alunos, eu falo que é um tapete de colágeno, onde são precipitados cálcio e fosfato para a gente formar os cristais de hidroxiapatita. E com relação às células, a gente tem aqui os osteoblastos. Tá? I'm trying to manage this. A gente tem os osteoblastos, a gente tem as bone lining cells, a gente tem também os osteócitos, que basicamente fazem parte da mesma família. Então, são células de origem mesenquimal. E aí a gente tem os osteoclastos, que vão ser aquelas células que têm a origem hematopoética e que são responsáveis pela reabsorção do tecido ósseo. Os 3 anteriores que eu falei, eles são responsáveis pela síntese de matriz orgânica. Então, eu vou falar um pouquinho de cada um deles. Osteoblastos e bone lining cells, então, são células que são responsáveis pela formação de todas aquelas proteínas que constituem a matriz orgânica do tecido ósseo. Na verdade, a gente consegue avaliar, então, os osteoblastos a partir da expressão de algumas proteínas que vão ser muito importantes, que caracterizam os estágios de desenvolvimento dele. Então, a gente tem, por exemplo, a RANX2, que mostra para a gente aquela passagem do pré-osteoblasto para o osteoblasto. E depois, quando ele está numa fase mais ativa funcional, ele começa a produzir uma série de proteínas que são aquelas que eu mostrei para vocês, que constituem a matriz orgânica do tecido ósseo. Então, aqui a gente tem um esqueminha só mostrando as coisas que são produzidas pelos osteoblastos. E aí a gente tem fatores de crescimento, que são muito importantes, o colágeno tipo 1, aquelas outras proteínas não colágenas, a fosfatase alcalina, e o intuito é sempre sintetizar a matriz orgânica. Lembrando que, a partir do momento que o osteoblasto ele deixa de estar ativo, ele se transforma em bone lining cells, que são aquelas células que eu tinha mostrado anteriormente. A partir do momento que há necessidade, ele volta a ser um osteoblasto em atividade e volta a ter a síntese da matriz orgânica. Os osteócitos são aqueles osteoblastos que ficam aprisionados na matriz orgânica. E eram células que, até um tempo atrás, eles eram muito negligenciados, porque pensava-se que os osteócitos eram um osteoblasto que parou de ter atividade e ele simplesmente ficava lá preso naquela matriz do tecido ósseo. Hoje em dia já se sabe que eles são super importantes para a mecanotransdução, porque, na verdade, eles são considerados mecanotransdutores. A mecanotransdução é um termo que a gente pegou emprestado, né, da engenharia, em que a gente fala que a mecanotransdução é a conversão de sinal. Então, a gente transforma sinais mecânicos, sinais físicos, em sinais químicos. Como é que é feita essa leitura? A gente aplica um estímulo no tecido ósseo, o osteócito detecta isso, e aí ele converte esse estímulo em substâncias químicas. Por exemplo, aqui, olha, eu tenho a SOST, né, a esclerostina, que é um dos exemplos. E esses sinais químicos eles vão estar trabalhando para que a gente tenha formação ou reabsorção de tecido ósseo. Então, na verdade, hoje em dia, já se sabe que o osteócito é praticamente o controlador de todo o equilíbrio do tecido ósseo, porque ele começa com essas projeções que a gente observa aqui, nesse corte de desgaste, a gente vê que esses osteócitos eles estão em íntima comunicação e, na verdade, os estímulos que são detectados num local acabam sendo transferidos para outro local. E a gente tem essa produção de substâncias que vão provocar formação ou reabsorção óssea. E aí, como é que isso acontece, né? Na verdade, a gente tem... aqui seria um esqueminha mostrando como é que seria o tecido ósseo com a presença dos osteócitos. Eles têm um fluido ao redor dos seus canalículos. E, a partir do momento que a gente tem uma carga, a gente tem um estímulo, load, on osteocytes, a gente tem uma conversão feita por integrinas, que são receptores transmembrana, e aí eles vão ativar sinalizações aqui no interior dos osteócitos, de tal forma que a gente tenha a síntese de todas essas substâncias, dependendo da aplicação do estímulo ou da não aplicação do estímulo. E aí eu quero chamar a atenção de vocês para a esclerostina, porque, na verdade, ela é uma proteína que ela tem sido bastante estudada agora, porque ela está intimamente relacionada com a via 20, que é uma via que a professora Ana estuda, né, nos projetos dela, junto com a beta-catenina, que é uma das principais vias para a formação de tecido ósseo hoje em dia. Então, vou só falar um pouquinho mais dela. A via 20 é uma via que ela foi descrita a partir de 2007, mais ou menos, e ela é uma das principais vias relacionadas com o processo de formação óssea. Na verdade, quando a gente fala na via 20, ela tem 3 vias de atuação. We have 3 signaling pathways. Mas a mais importante para a gente no tecido ósseo é a via canônica, que é a via 20 beta-catenina. E aí, na verdade, quando a gente tem a ligação, a ativação dessa via 20 beta-catenina, em que a gente tem a ligação aqui junto ao receptor, a gente consegue ter um processo de formação óssea. E isso é muito importante para que a gente consiga manter toda essa atividade de síntese da matriz orgânica do tecido ósseo e a precipitação de minerais. Como é que entra a esclerostina nesse sentido? A esclerostina ela vai atrapalhar a ligação ou a ativação da via 20 e, dessa forma, ela é uma inibidora do processo de formação óssea. Tá? Então, por isso que ela está sendo bastante estudada. E, na verdade, o que se pensou é que, quando a gente tem a ligação da 20 com a ativação da via 20, na verdade, a gente tem um processo de formação óssea, foi detectado, na verdade, que quando se observava uma inibição ou a ausência da expressão da esclerotina, que seria o antagonista, observava-se situações em que se tinha esclerose óssea. E aí, no caso, aqui é um esqueminha, na verdade, que eu peguei aqui de um paper, né? E que está falando, na verdade, que aqui a gente tem o processo de formação óssea. E, na verdade, a esclerotina bloqueia esse processo. Quando se observava que tinha alguma alteração gênica em que a esclerotina não se expressava, a gente observava condições de esclerose óssea. Por que eu estou falando isso para vocês? Porque dentro dos tratamentos para osteoporose, uma das coisas que está se estudando agora, que estão em clinical trials, é justamente um antagonista da esclerotina, porque seria exatamente isso que a gente mostrou aqui para vocês. A gente inibe a esclerotina, libera a via 20 e você tem um estímulo para a formação óssea. Esse clinical trial é o Frame Study, né? A medicação chama romemozumab, e ela está em estudo nos Estados Unidos, porque ela é um potencial medicamento para o tratamento de osteoporose. Tá? Então, só queria falar um pouquinho dela, porque, na verdade, isso é uma coisa bem recente mesmo. Por último, a gente tem os osteoclastos, que são células hematopoéticas, na verdade, são macrófagos, macrophages, que fazem a reabsorção óssea. Então, eles têm a capacidade de se ligar ao tecido ósseo, eles se fixam lá, e aí eles começam a fazer a reabsorção óssea a partir de uma série de enzimas, né? Que, na verdade, a gente tem aqui a presença do rank ligante, que ativa os pré-osteoclastos. A gente tem a TREF, a catepsina K, a gente tem a TRAP, que eu coloquei aqui. Então, todos esses são sinalizadores de uma atividade osteoclástica. E ele faz o contraponto. Enquanto a gente tem a formação pelos osteoblastos, todas aquelas linhagens mesenquimais, a gente tem os osteoclastos fazendo a reabsorção óssea. E, com relação a essa via para a reabsorção óssea, em 1997 foram descritos os novos membros dos fatores de necrose tumoral. O sistema OPG rank ligante, que, na verdade, é o sistema que está mais relacionado a essa via de reabsorção e ativação osteoclástica. Então, aqui a gente tem que o osteoblasto ele sintetiza rank ligante e OPG. O rank ligante ele se liga a rank e, dessa ligação, a gente ativa os osteoclastos. No entanto, se o OPG desloca a rank ligante, essa via não é ativada e a gente tem um processo de formação óssea. Portanto, esses novos membros do fator de necrose tumoral eles são muito importantes quando a gente pensa nessa caracterização dessa remodelação óssea, formação e reabsorção óssea. E, juntando tudo isso que eu falei para vocês com relação a essa atividade de osteoblastos, osteócitos, bone lining cells e osteoclastos, a gente tem, então, as BMUs, que seriam as unidades multicelulares básicas compostas por todas essas células que vão estar fazendo sinalizações e mantendo o tecido ósseo em equilíbrio. E aí a gente consegue, então, manter a dinâmica dele. E isso vai ser importante para fazer correção de microfraturas, também é importante para orientar a massa corporal do tecido ósseo, adequar essa massa óssea em resposta aos estímulos mecânicos e para manter a homeostasia mineral. Bom, então essa é uma situação que, na verdade, é fisiológica, né? Nós temos, então, aquela situação em que a gente tem essa dinâmica do tecido ósseo que vai ser comandada por cada uma dessas células e algumas dessas vias de sinalização que eu apresentei para vocês. Na verdade, é o PG rank L e também a via 20, uma de reabsorção e uma de formação. Em situações em que a gente tem algum estímulo, alguma agressão, alguma cavidade, alguma situação que você tenha que ter um reparo ósseo, a gente tem algumas etapas que precisam acontecer para que a gente consiga ter essa reparação acontecendo adequadamente. A gente começa com uma resposta inflamatória, depois a gente tem a formação de um tecido mineralizado e depois a gente tem a resposta de remodelação óssea. Na verdade, aqui a gente tem, então, histologicamente, uma resposta inflamatória e uma formação de um coágulo sanguíneo, um tecido de granulação, que também ainda faz parte dessa resposta inflamatória. Daí a gente tem a síntese de uma matriz colágena e a mineralização, e depois a remodelação óssea. E essas são as etapas que acontecem quando a gente tem um processo de reparo ósseo acontecendo após alguma agressão. E são esses os modelos que a gente utiliza para estudar no nosso laboratório. A gente utiliza um modelo que são os ratos, e a gente utiliza a exodontia do incisivo superior de rato. É o upper incisor extraction. We extract the upper incisor, and then we wait for the time to repair that dental socket. A gente faz a extração e depois a gente espera um tempo para que aconteça essa reparação. Approximately 28 days, aproximadamente 28 dias, in normal conditions, em condições normais. Aqui esse é um processo em que a gente tem um instrumental adaptado, o dente é extremamente curvo, né? Aqui ele está reparado, e aqui eu só coloquei umas amostras para vocês verem como é que ficam na microtomografia, como é que é aquele alvéolo reparado. Agora eu tenho aqui alguns resultados que eu vou mostrar para vocês. Esse foi um trabalho que a gente publicou recentemente. A gente colocou aqui o QR Code para quem quiser baixar ele depois, tá? Tem aqui para vocês baixarem. Então, nesse estudo, a gente fez um estudo do processo de reparo alveolar, mas a gente fez uma abordagem imunohistoquímica, microtomográfica e também molecular. Na verdade, esse processo já estava descrito desde a década de 60. Né? A gente tinha toda essa cronologia muito bem descrita, mas agora a gente queria utilizar essas outras ferramentas para a gente ver efetivamente como é que as coisas estavam acontecendo lá no interior do alvéolo. E aí, uma informação muito interessante que a gente teve é que aos 14 dias, a gente estudou 7, 14 e 28 dias. 7, 14 and 28 days after the tooth extraction. E a gente observou que aos 14 dias a gente tinha uma expressão muito importante da TRAP, que é um marcador da atividade osteoclástica. E, na verdade, quando nós fizemos análise por biologia molecular e a gente fez a expressão gênica relativa para OPG e rank-L, e aí a gente fez a proporção de rank-L por OPG, marcando a taxa de reabsorção, a gente observou que, com 14 dias, essa resposta estava aumentada. Tá? Na verdade, aqui, então, seria a ativação para que a gente tenha essa atividade osteoclástica. A gente anteriormente achava que essa atividade osteoclástica ela estaria aumentada apenas próximos aos 28 dias. Na verdade, o que a gente observou é que aos 14 dias aquela BMU já começa a entrar em atividade e a gente já tem aquele turnover ósseo acontecendo no interior do alvéolo. Então, essa foi uma informação bastante interessante que a gente obteve com essas novas ferramentas. E, além disso, a gente fez também algumas análises por microtomografia e aqueles parâmetros tradicionais. A gente observou, então, volume ósseo aumentado com o passar do tempo, fração de volume ósseo também aumentado, o trabeculado ósseo aumentando a sua espessura e a porosidade total diminuindo. Então, a gente observou que, realmente, todos aqueles processos na microarquitetura óssea eles estavam acontecendo de acordo com o esperado. E o que a gente inseriu depois nesse contexto? A gente tem que pensar que, depois, com o passar do tempo daquele alvéolo reparado, a gente tem processos de reabsorção que fisiologicamente acontecem. E é nessa situação que o paciente vem para que ele possa ser submetido aos procedimentos reabilitadores. E, junto com isso, a gente tem também algumas interferências sistêmicas que esse paciente pode apresentar. Então, eu até coloquei aqui uns questionamentos. Né? A gente tem a dinâmica do tecido ósseo, ou a remodelação óssea, que é um processo natural, em que a gente observa essas perdas alveolares que vão acontecendo com o passar do tempo. Como seria o comportamento do osso durante o processo de reparo em situações em que a gente tem as interferências sistêmicas? Pacientes osteoporóticos, diabéticos, hipertensos e por aí vai. E, além disso, pacientes que fazem uso de medicações para tratarem essas interferências sistêmicas, eles conseguem melhorar o seu reparo ósseo? Então, na verdade, são esses os questionamentos que a gente utiliza para desenvolver as pesquisas no nosso laboratório. E aí, a partir daí, eu vou mostrar para vocês alguns trabalhos que a gente tem desenvolvido. Tá? Esse trabalho ele saiu recentemente e a gente começou a trabalhar com um modelo de obesidade, em que esses animais eles eram alimentados com uma dieta que ela aumentava, era hipercalórica, né? E aí a gente quis observar como é que estava o processo de reparo alveolar desses animais. Então, foi feito a exodontia, nessa condição em que eles estavam obesos. Aqui a gente tem uma tabela em que a gente vê realmente que ele está com todos aqueles parâmetros, o índice de Lee, a massa corporal, estava tudo alterado. E aí nós fizemos a exodontia e avaliamos aos 7, 14 e 28 dias. Aqui a gente tem o animal controle e aqui o animal com aumento de peso. Com relação à expressão da TRAP, e esse foi o dado mais interessante, a gente observou que nos animais com aumento de peso, aos 7, 14 e 28 dias, a gente tem uma maior expressão da TRAP em relação ao controle, mostrando que a gente tem um aumento dessa resposta de reabsorção, muito provavelmente por conta desse link com a resposta inflamatória, que seria por causa dos macrófagos. Isso está respondido? Yes, please. Wait. Not eating, they are eating the same, but the diet that was produced with casein, I don't remember what was the substance that were produced in the diet, but it was high calories. Yes. So they eat this, but it's very interesting you asking, because we have to give this, and the animals, they got, I don't know how to say, saciados. They were full, yes, with a small quantity of the diet. But we have to give something that make him like to eat the food. So we put sausages, we put Oreo, the things, but everything was part of the protocol. And then they have to eat more, not more, the same as the other, but because he felt full and then he stopped to eat. And we need that they eat the same quantity, okay? Nessa condição, a gente observou, então, que tinha uma expressão muito grande de TRAP nos 3 períodos. E isso, na verdade, foi explicado pela expressão de Ranchielli-OPG. A gente observa que Ranchielli-OPG, que são os marcadores para reabsorção óssea, ou seja, eles sinalizam para que aconteça a reabsorção. Então, eles estavam, a Ranchielli também aumentada nos animais obesos em relação à OPG, tá? Nos animais obesos. Então, se a gente observar aqui a comparação, a gente vai ver que pela área de imunomarcação, a gente tem uma maior marcação da Ranchielli em relação à OPG nos 3 períodos nos animais obesos. Além disso, a gente marcou também a osteocalcina, que é uma proteína que marca a mineralização óssea, que ela é importante para a gente ver os osteoblastos que estão maduros e sintetizando uma matriz mineralizada que se precipita, né, sobre a matriz colágena. E aí nós observamos também que a gente tinha uma expressão adequada, né, da osteocalcina, aumentando com o passar do tempo, e isso não foi observado nos animais obesos. Com relação à VEGF, foi um dado importante, porque a VEGF é um fator que mostra para mim a osteocondução. Seria o processo em que a gente tem a produção de vasos para que a gente melhore a formação óssea. E a gente observa que há um atraso nessa resposta, porque o que eu vejo da VEGF aqui aos 14 dias no animal controle, eu vou ver aqui aos 28 dias no animal obeso. Então, o processo acontece, mas acontece um pouquinho mais atrasado. There's a delay in the expression of VEGF in the obese animals. Okay? Então, o que nós observamos é que a obesidade é uma situação em que realmente ela atrasa o processo de reparo alveolar e, portanto, tem um impacto, né, nesse ganho de peso com relação aos tratamentos dentais que podem ser propostos para o paciente. Só abrindo um parênteses, a gente tem trabalhado com esse modelo agora, o projeto que a gente está em desenvolvimento no laboratório é o de síndrome metabólica, metabolic syndrome. And when we put this diet, and we made the animals become osteoporotic. So in this condition, we are trying to study the bone repair. Okay? Nós gostamos de dar umas comidas diferentes para os ratos. Então, agora, a gente começou a trabalhar também com o modelo de diabetes tipo 2, type 2 diabetes. E nessa situação, a gente também buscou um protocolo em que a gente dá uma dieta. O nome dessa dieta é dieta de cafeteria, cafeteria diet. A gente tem toda essa receitinha aqui, né? Bolacha recheada, bolacha wafer, salgadinho de milho, mais água com açúcar. A gente dá para os animais 3 semanas, 3 weeks. que eles recebem essa dieta. In the 4th week, na 4ª semana, a gente dá uma injeção de streptozotocina, baixa dose, low dose of streptozotocina. And this condition, streptozotocina é uma droga que ela causa, ela destrói as células beta-pancreáticas, que são as responsáveis pela produção de insulina. Então, a gente dá uma dose baixa, e aí o animal ele se torna diabético tipo 2. Uma glicemia de 150, 180 miligramas por decilitro, que é um modelo que a gente quer estudar mesmo, que é o modelo que mais a gente observa na população. Então, esse foi o projeto de iniciação científica da minha aluna, da Letícia, que ela finalizou agora. Nós fizemos um modelo de reparo alveolar e a gente observou marcadores da resposta inflamatória. E a gente viu que nos períodos iniciais, aos 3 e 7 dias, a gente vê que nos animais diabéticos a gente tem uma maior expressão desses marcadores inflamatórios do que nos animais controle. Um outro marcador que foi o TNF, também a gente observa que ele estava mais aumentado nos animais diabéticos em relação ao controle. Aqui, aos 7 dias, a gente já consegue observar um começo de organização do trabeculado ósseo nos animais controles. TGF também mais expressa nos animais diabéticos em relação ao controle. E isso é importante só chamar a atenção de vocês que a inflamação faz parte da resposta reparacional. Então, ela precisa acontecer. No entanto, ela estava exacerbada nos animais que eram portadores do diabetes tipo 2. A gente fez essa análise com 3 e 7 dias. A partir de 14 e 42 dias, nós avaliamos as proteínas ósseas. Então, nós avaliamos a 20 e nós observamos que agora inverteu. A gente tem uma menor expressão da 20 em comparação aos animais controles, em que a gente tem uma maior expressão. A marcação é marrom, brown. Então, todas essas áreas marrons, a gente tem a marcação positiva para as proteínas. A osteocalcina, ela estava mais marcada aqui nos animais controle, e aqui a gente tem um trabeculado ósseo mais organizado em relação aos animais diabéticos. Com relação à TRAP, a gente observa que nós temos aqui a presença da TRAP, no entanto, nos animais diabéticos, a gente tem uma maior expressão em relação aos animais controles nesses períodos mais tardios. Portanto, a gente observa que, com relação à expressão das proteínas da matriz, a gente vê que realmente há uma situação favorável para uma resposta de reabsorção, em contraposição à resposta de formação, nessa condição de diabetes. E isso a gente conseguiu depois confirmar pelos dados microtomográficos, em que a gente vê que os animais controle, eles têm uma maior fração, percentual de volume ósseo, uma trabécula óssea mais espessa também, e aqui a gente vê que os diabéticos têm uma porosidade maior no trabeculado ósseo. Portanto, a gente observa que realmente há um prejuízo nesse processo. Esse trabalho tem outras análises. A gente avaliou também a maturação de colágeno, a gente avaliou a dinâmica do tecido ósseo, só que eu não trouxe os resultados aqui. Mas todos eles apontam para essa melhora do reparo nos animais controle em relação aos diabéticos. E a grande menina dos nossos olhos, que é a condição de osteoporose, que é aquela situação em que a gente tem, né, as pessoas, a partir de uma certa idade, com a perda dos hormônios, né, e, no caso das mulheres, quando atingem a menopausa, é uma condição que acomete bastante a população. Portanto, a gente tem um olhar mais cuidadoso para isso. O nosso modelo experimental é a ovarectomia bilateral, bilateral ovarectomy. A gente remove, então, os ovários dessas ratas, tem todo um cuidado, é todo um protocolo que é feito. E como a nossa preocupação é em contornar o animal osteoporótico, mas depois também tentar tratá-lo e ver como é que fica essa resposta frente aos tratamentos medicamentosos, a gente propõe alguns tratamentos medicamentosos que eu vou estar apresentando aqui para vocês. São medicamentos, na verdade, que a gente tem utilizado, que estão disponíveis comercialmente, e a gente adapta eles para o tamanho dos animais, e aí a gente administra para observar como é que é o comportamento do osso reparacional. Então, aqui, só mostrando para vocês que esses tratamentos ou a gente faz por injeção subcutânea ou por gavagem. Tá? Então, são sempre assim que a gente vai estar desenvolvendo os nossos projetos. E aí a gente tem uma série de fármacos que podem ser estudados, né, por conta do tratamento da osteoporose. O primeiro trabalho que a gente começou a fazer, a gente estudou uma medicação que é um modulador seletivo de receptor de estrógeno, é o haloxifeno. Esse medicamento ele é bastante interessante porque ele tem uma seletividade para os receptores de estrógeno que estão presentes nos osteoblastos. Portanto, ele tem uma atração pelos receptores de estrógeno no osteoblasto. No entanto, eles não vão ter nenhuma afinidade com os receptores presentes na mama, no ovário e por aí vai. Então, na verdade, por isso que ele é um modulador seletivo. É um selective estrogen receptor modulator. This is the medicine that we tried. E nesse primeiro estudo, a gente comparou ele justamente com a reposição hormonal, para ver como é que era o comportamento dele junto à reposição hormonal, que é o padrão ouro para o tratamento de osteoporose. E o que nós observamos aqui, ao longo do processo de reparo alveolar, e aqui, aqui a gente tem, então, 7, 14, 21, 28 e 42 dias. A gente tem aqui na primeira coluninha o animal chã, a osteoporótica, a tratada com estrógeno e a tratada com haloxifeno, sempre nessa sequência. A gente observa que os melhores resultados realmente foram observados com as ratas chã ao longo do tempo, sempre a coluna branca é a maior. E a gente observa que, com relação às terapias que a gente propõe, inicialmente, a reposição de estrógeno, que é a barra preta, ela tem um resultado melhor do que o haloxifeno, que é a última barra. No entanto, no final do tratamento, elas já estão equivalentes. Então, a longo prazo, o haloxifeno acaba tendo um resultado bem semelhante à reposição hormonal. Com relação à espessura do trabeculado ósseo, que foi a outra análise que nós fizemos, a gente observa que tanto o chã, como o estrógeno, como o haloxifeno, têm respostas semelhantes e superiores aos animais osteoporóticos. Com relação à marcação de osteocalcina, nós observamos aqui que a expressão da osteocalcina, aos 14 dias, ela estava maior no chã e no estrógeno e no haloxifeno elas estavam bastante semelhantes. Tá? Então, a gente observa que, realmente, com relação à expressão desse marcador, a gente vê que o comportamento acaba sendo muito semelhante nesses 3 grupos e sempre superiores aos animais osteoporóticos. Portanto, essa terapia que a gente propôs com o haloxifeno e a reposição hormonal, elas realmente conseguiram tornar o reparo ósseo semelhante aos animais chã e conseguiram manter uma expressão de osteocalcina também semelhante aos animais chã. A osteocalcina, que é um marcador da mineralização óssea. A gente fez um outro estudo, em que a gente utilizou também esses mesmos grupos, mas a gente fez uma marcação concomitante por imunofluorescência ou colocalização de OPG e Ranchieri. E aí a gente observou, foi também um dado interessante, que aos 7 dias, a gente tem uma marcação de OPG e Ranchieri semelhante nos animais chã e nos animais osteoporóticos. No entanto, depois, com o passar do tempo, essa marcação de OPG e Ranchieri ela vai diminuindo nos animais tratados, nos animais osteoporóticos. E aqui no gráfico, quando a gente vê o conjunto total dos resultados, a gente vê que, no final, que seriam 28 e 42 dias, o turnover ósseo, que seria essa expressão conjunta de OPG e Ranchieri, ela está semelhante nos animais chã, que seria a primeira coluna, e dos animais haloxifeno, que seria a última coluna para cada período analisado. E o último trabalho que a gente fez nessa série, a gente avaliou apenas a atividade osteoclástica com esses 4 grupos experimentais. E a gente observou que, realmente, nos animais tratados com haloxifeno, a gente tem uma diminuição na expressão de TRAP, ou seja, uma menor atividade de reabsorção óssea. Por isso que essa medicação é um potente antirreabsortivo. No entanto, ele tem um efeito bastante fisiológico por essa seletividade pelos receptores apenas dos osteoblastos. Isso fez com que a gente depois optasse por continuar estudando essa medicação em outros estudos que eu vou mostrar mais para frente. Que já é nesse aqui, em que a gente fez alguns estudos com drogas antirreabsortivas, mas a gente comparou agora o haloxifeno, que era a medicação que deu o melhor resultado, em comparação com o alendronato, que é um bifosfonato oral. São duas vias de atuação diferentes. Enquanto o alendronato ele causa apoptose osteoclástica, porque ele inibe aquela via do mevalonato e causa a apoptose, os osteoclastos não conseguem se manter ativos. Essa é a via dele como um antirreabsortivo. O raloxifeno é um modulador seletivo de receptores de estrógeno. O que a gente observou nesse estudo, e eu vou aqui direto para o gráfico, e aqui a gente avaliou só 14 e 42 dias. Primeiro aqui seria o rato com alendronato, depois é o rato osteoporótico, depois é o rato com raloxifeno e o último rato chã. Aos 14 dias, a gente vê que os resultados são bastante semelhantes, mas a gente tem valores numéricos maiores para o tratamento com alendronato. No entanto, quando a gente passa para 42 dias, a gente vê que os maiores valores foram observados nos animais chã e nos animais tratados com raloxifeno, mostrando, então, que a longo prazo o raloxifeno mostrou-se melhor do que o alendronato para melhorar o processo de reparo alveolar em ratos. Ainda, um outro estudo que a gente tem desenvolvido, e esse saiu recentemente também, é fazendo o tratamento de teriparatida de forma intermitente. Porque, na verdade, a aplicação da teriparatida de forma intermitente nos animais, ela causa uma ativação da via 20 e, consequentemente, a gente tem uma resposta de formação óssea. A teriparatida ela é o único anabólico ósseo que ele é aprovado para utilização pela FDA. Tá? Então, com doses intermitentes, a gente, nesse caso, a gente fez a castração. We performed the orchiectomy of the animals. So now we are looking for the osteoporosis, but in male, not in female. Então, aqui a cirurgia que os alunos fizeram. E aqui a gente observou a expressão de proteínas da matriz ao longo do processo de reparo alveolar. Aqui são os animais chã. Aqui a gente tem os animais orquiectomizados, osteoporóticos, e aqui tratados com a teriparatida. E a gente observa que aos 14 dias, a gente tem eu vou falar aqui apenas da teriparatida, uma importante expressão dos marcadores de formação óssea e menor de reabsorção, em comparação até aos grupos anteriores, inclusive o chã. Passando para os 42 dias, mais uma vez a gente observa uma maior expressão dos marcadores de formação óssea e uma menor de reabsorção em comparação aos grupos anteriores. Lembrando que aqui nós avaliamos a 20, que é aquela via de formação que ela estava ativada pela teriparatida intermitente, a fosfatase alcalina, que controla a precipitação de fosfato, a oxocalcina, que é um marcador de mineralização óssea, e a TRAP, que faz a reabsorção óssea ou marca a atividade osteoclástica. Então, a gente observou que, nesse modelo experimental, a injeção intermitente da teriparatida ela foi eficiente realmente para promover uma melhora no reparo alveolar. E aí eu coloquei alguns parâmetros também de microtomografia, em que a gente observa que uma fração de volume óssea maior no grupo para teriparatida, assim como uma maior espessura do trabeculado ósseo. A teriparatida que a gente administrou nos animais é a Fertel, teriparatide, injeções intermitentes, né? Então, todos os dias a gente injetava uma pequena concentração para os animais, e ela realmente respondeu tendo um efeito como um anabólico de tecido ósseo. Não sei se vocês conhecem, é uma substância que se chama OPGFC. Existe uma medicação que ela é chamada de Prolia, que é uma medicação que ela tem sido muito utilizada para o tratamento da osteoporose, e os médicos têm optado bastante por ela, porque são injeções a cada 6 meses. Então, para a paciente é uma coisa um pouco mais tranquila, porque só tem que tomar 2 injeçõezinhas por ano. Para a gente estudar essa medicação, a gente não consegue utilizar, porque ele é um anticorpo monoclonal da Ranquiel, a gente não consegue utilizar isso nos ratos, então a gente precisou comprar o PGFC. This is OPGFC, is the OPG associated to a complement fragment. Complement, yes, complement. And then this is similar to denosumab. Denosumab is an anti-osteoporotic drug that has an important effect anti-resorptive. Então, a gente trabalhou com ele, a gente observou que a OPGFC realmente foi importante para não deixar que a gente tivesse a expressão de Ranquiel no plasma. E o que nós observamos aqui no processo de reparo alveolar foi que realmente ele foi potente como um antirreabsortivo, quando a gente fez uma análise por fluorocromos. Aqui, essa análise por fluorocromos é uma análise em que, quando a gente faz a injeção de um fluorocromo verde, essa expressão verde me mostra o osso antigo, e o vermelho me mostra um osso mais recente. Então, no tratamento com a PGFC, a gente observa que tem uma grande expressão do verde em relação ao vermelho, mostrando que ele estava realmente tendo uma importante atividade antirreabsortiva. Havia também uma precipitação mineral diária que estava semelhante aos animais Cham para o PGFC. No entanto, essas foram as únicas respostas favoráveis que a gente observou no metabolismo do osso reparacional. Nos demais parâmetros que a gente observou, a OPGFC ela não conseguiu melhorar a qualidade da microarquitetura do tecido ósseo. Então, esse é um fármaco que realmente a gente precisa prestar um pouquinho de atenção. Ele está sendo muito utilizado e ele não consegue cumprir completamente o seu papel com relação a inibir a reabsorção, que isso ele faz, mas depois estimular uma formação óssea adequada, né? Então, por isso que vai o alerta, a gente vai continuar estudando essa medicação no laboratório. Mas aí a gente saiu um pouquinho desse modelo de reparo alveolar e a gente passou agora para o reparo periimplantar, que aí, nessa situação, a gente instala os implantes. No entanto, a gente instala esses implantes na tíbia dos ratos, mas a gente avalia apenas a região medular, que é a região onde o turnover do tecido ósseo é semelhante ao turnover do osso alveolar. Então, aqui a gente tem... Esse é um paper que a gente se baseia para utilizar esse modelo, em que a gente instala na tíbia, mas a gente tem como área de interesse apenas a região da terceira, quinta espira, que seria a região onde a gente tem o osso medular, que seria o turnover semelhante ao osso alveolar. Aqui é o implante sendo instalado. Aqui, olha só, para vocês verem como é que fica quando a gente faz a injeção com os fluorocromos. E aí a gente tem alguns estudos também que a gente desenvolveu. Então, esse trabalho aqui, a gente fez uma comparação entre haloxifeno e alendronato, mas agora numa condição de osteoporose e após a instalação de implante. Então, agora foi um reparo periimplantar ao invés de reparo alveolar. E aqui nas nossas análises, eu queria chamar a atenção de vocês. Aqui, a gente observou essa região aqui, onde está em azulzinho, que é a região de interesse, que é a região onde a gente teria o osso medular. É nessa região que a gente olha, que é a nossa região que a gente faz a análise, porque é nessa região que a gente tem o reparo turnover semelhante ao osso alveolar. E aí, o que a gente observou aqui é que a gente tem alguns parâmetros que mostram para a gente, principalmente aqui, ó, com relação à quantidade de volume ósseo, número de trabéculas. Eu vou falar para vocês aqui a sequência. Ó. A gente tem aqui o rato osteoporótico, Cham, com alendronato e com haloxifeno. É essa sequência que está aqui também, osteoporótico, Cham, alendronato e haloxifeno. A gente observa que o alendronato ele consegue ter uma quantidade de tecido ósseo maior. No entanto, quando a gente observa que nos cortes histológicos, que a gente tem aqui um corte feito em tecidos calcificados, a gente observa que no alendronato a gente tem uma quantidade muito grande de medula, diferente dos animais Cham e dos animais com haloxifeno. Essa resposta também ficou bastante interessante para a gente, porque a gente foi fazer uma análise com os fluorocromos, em que a gente faz a injeção de marcadores ósseos fluorescentes em momentos diferentes, marcando a precipitação de cálcio sobre a matriz. E aí a gente faz uma injeção com verde, que é a calceína, e com vermelho, que é a lisarina. Verde, marcando o osso antigo, vermelho, marcando o osso mais novo. A gente consegue fazer essa injeção nos mesmos animais, portanto, quando a gente faz a análise, a gente consegue observar o osso mais novo e o osso mais antigo. E o que nos chama a atenção é que, realmente, o grupo alendronato ele tem uma maior quantidade óssea. No entanto, é uma maior quantidade de osso antigo. Portanto, é um osso que ele não tem um turnover tão adequado, ele realmente tem uma resposta antirreabsortiva, e aquele osso que permanece lá não é um osso tão funcional. Tá? E por esse motivo que a gente não tem um reparo ósseo tão adequado. Aqui a gente fez algumas análises marcando a área de osso antigo e osso novo. A gente observa aqui, em verde, a quantidade de calceína, e em vermelho, a quantidade de alizarina. Aqui seria sham, osteoporótico, haloxifeno, alendronato e haloxifeno, desculpa. E essa análise a gente faz também com relação à taxa de precipitação mineral diária. E a gente observa que a maior taxa de precipitação mineral diária foi observada nos animais sham e nos animais haloxifeno. Já as menores taxas foram observadas nos animais osteoporóticos e também nos animais tratados com alendronato. Portanto, o que a gente conseguiu verificar é que realmente tem uma quantidade grande de tecido ósseo nos animais tratados com alendronato. No entanto, é um osso que não é tão vital. É mais um osso antigo e a gente não tem uma taxa de renovação igual a que a gente observou nos animais tratados com haloxifeno. Isso a gente observou tanto para o alendronato e haloxifeno, tanto para o alvéolo, como também para os implantes. E aí a gente fez um outro estudo, em que a gente quis agora avaliar a biomecânica, que seria a remoção dos implantes para saber se ele tinha uma maior ou uma menor osseointegração. E aí a gente observou que quando a gente fez o contratorque, que seria a remoção daqueles implantes, e a gente avaliou a força necessária para remover esses implantes, a gente viu que nos animais sham a gente teve os maiores valores, seguido dos animais tratados com haloxifeno, e os menores valores foram para os animais osteoporóticos e tratados com alendronato. Portanto, é um osso maior, em maior quantidade. No entanto, é um osso que tem menos vitalidade, porque é um osso mais antigo, e isso se confirma nos testes biomecânicos, porque a gente vê que o torque de remoção foi menor. O maior torque de remoção foi o observado para os animais tratados com haloxifeno e também nos animais sham. Tem duas colunas, porque, na verdade, a mais clara representa implantes de superfície usinada e a mais escura representa os implantes de superfície tratada com duplo ataque ácido. Aí, a partir desse trabalho, agora a gente trabalha com os implantes apenas de superfície de duplo ataque ácido. Então, com relação às nossas medicações, a gente viu que, em comparação ao haloxifeno e ao alendronato, a gente viu que ele acaba tendo um resultado melhor para a qualidade do osso reparacional, tanto após a exodontia, como também após a instalação de implantes. Uma outra situação que a gente estudou no laboratório é a condição de animais que são pinealectomizados. Para quem não sabe, a pineal é uma glândula que ela é responsável pela síntese de melatonina, que é uma substância que controla o nosso ritmo circadiano. Então, na verdade, ela, em oposição ao cortisol, ela faz um controle para o nosso ciclo sono-vigília, para que a gente consiga, então, ter todas as etapas, né, do dia acontecendo certinho, que a gente tem à disposição. A melatonina é o hormônio do sono, que a gente fala. Para ele ser produzido, na verdade, a gente precisa estar no escuro e na ausência de barulho. E a gente precisa ter essa produção de melatonina, porque o nosso organismo precisa dela para a gente manter todas as funções adequadas. Na verdade, eu coloquei esse resultado aqui, esse trabalho aqui, porque eu sei que tem bastante gente que faz uso de melatonina. Não sei se aqui... Melatonina. I don't know if you... Yes? Então, as pessoas usam melatonina, geralmente, para dormir melhor, então, por isso que eu trouxe esse resultado. Nesse trabalho aqui, nós removemos a pineal dos animais. Então, aqui a gente coloca os animais no estereotáxico, aí a gente remove a glândula pineal, e aí a gente fez a instalação dos implantes. E aí, num outro grupo, a gente removeu a pineal, e aí a gente fez a gavagem diária de melatonina, sempre no mesmo horário, para simular aquela reposição exógena de melatonina. Tá? E aí a gente observou, com relação às proteínas da matriz, né, a gente viu que cada uma das proteínas que a gente avaliou, que foi a RANX2, fosfatase alcalina, também a ospontina e a oscalcina, são todos marcadores de formação óssea, a gente observou que nos animais pinealectomizados tratados com melatonina, a gente teve uma expressão dessas proteínas semelhantes ao grupo sham e superiores aos animais pinealectomizados, mostrando, então, que em nível de proteínas, em nível celular, a gente realmente tem um prejuízo e o tratamento com a melatonina parece ajudar a reverter esse processo. No entanto, a gente não conseguiu evidenciar isso também quando a gente olhou para os parâmetros microtomográficos. A gente viu que, na verdade, os resultados estavam bastante semelhantes nos grupos, tanto sham, quanto pinealectomizado, quanto tratados com melatonina. Tá? Apenas aqui, olha, na densidade de conectividade, que a gente viu que o tratamento com a melatonina acabou melhorando essa resposta. Tá? Mas já com a superfície ativa de mineralização, a gente observou que tanto o pinealectomizado como o tratamento com a melatonina, a gente viu que os resultados estavam muito semelhantes. Portanto, esse tratamento com a melatonina, ele conseguiu mostrar que a gente tem algumas respostas celulares, mas não foram suficientes para que a gente conseguisse expressar no osso que a gente estava avaliando. Faltou aqui a gente fazer uma análise nos animais chã tratados com melatonina. Isso é uma coisa que sempre questionam a gente, e eu acho que realmente faltou esse grupo, principalmente porque tem muitas pessoas que não faltam a pineal, né? No entanto, elas tomam a melatonina. Então, isso é uma coisa que a gente precisa voltar para o laboratório. A teriparatida, a gente também trabalhou ele no reparo ósseo periimplantar. E aí eu já vou passar direto, né, para os resultados. A gente observou que o tratamento com a teriparatida, que seria aqui essa última coluna, promoveu as respostas relacionadas à microarquitetura óssea muito semelhantes aos animais chã e superiores aos animais orquiectomizados apenas. Tá? Aqui são os parâmetros que a gente avaliou pela microtomografia, que é o volume ósseo, a porcentagem de volume ósseo, a espessura de trabéculas e a separação de trabéculas. Já com relação à porosidade e número de trabéculas, são fatores que mostram que essa microarquitetura não está tão favorável. Esses valores foram observados nos animais orquiectomizados. Então, realmente, a teriparatida ela mostrou também o seu papel bastante favorável para o metabolismo ósseo. Fazendo os estudos com relação aos fluorocromos, a gente observou aqui que, com o tratamento com a teriparatida, a gente tem realmente uma maior formação óssea em comparação ao grupo em que foi apenas orquiectomizado. E com relação à taxa de aposição mineral, apesar de não ter diferenças estatísticas com o grupo chã, a gente vê que os valores numéricos foram maiores e muito maiores em relação ao orquiectomizado. Essa análise é muito interessante porque ela me fala como é que está a taxa de precipitação mineral por dia. Tá? A gente só consegue fazer isso pelos fluorocromos, e aí isso eu acho que é muito importante porque fala para a gente como é que está o grau de mineralização do tecido ósseo. Portanto, a teriparatida é um fármaco realmente que é bastante interessante, ele está cumprindo o seu papel como anabólico ósseo e está sendo bastante utilizado também para o tratamento de osteoporose. Tá? Então, essa foi a conclusão que a gente teve e veio muito ao encontro dos resultados que a gente teve no reparo alveolar. E a última interferência sistêmica que eu ia mostrar para vocês é a hipertensão. A gente trabalha com os ratinhos que são espontaneamente hipertensos. Então, eles nascem normotensos, depois da 3ª semana de vida, eles se tornam espontaneamente hipertensos. E é uma hipertensão que ela é como se fosse aquela hipertensão essencial, em que a pessoa não tem nenhum fator predisponente, mas ela se torna hipertensa. Como a gente trabalha também com a questão da terapia, a gente optou por tratar essa hipertensão com losartan, que é um fármaco que é um inibidor do sistema renina-angiotensina e é um dos mais utilizados pela população para fazer o tratamento para a hipertensão. E aí a gente fez esse trabalho, a gente avaliou a osseointegração, e aí eu sinalizei aqui os resultados mais importantes, porque aqui a gente viu que no tórax de remoção, o menor valor que a gente observou foi o animal espontaneamente hipertenso, que é a sigla SHR. No entanto, quando a gente fez o tratamento com losartan, ele conseguiu reverter aquele resultado desfavorável, fazendo com que o tórax de remoção obtivesse valores muito semelhantes aos animais normotensos. Então, realmente, o losartan ele foi eficiente em tentar reverter aquele prejuízo naquela resposta de osseointegração. E isso a gente confirmou também com alguns parâmetros microtomográficos, em que a gente observa que os menores valores foram nos animais espontaneamente hipertensos. No entanto, a partir do momento que eles foram tratados com losartan, acho que eu consigo mostrar melhor assim, a gente observa que aqui é o SHR, aqui é o tratamento com losartan. A gente observa que esses parâmetros eles foram melhorados quando a gente fez a terapia com losartan. Ele é uma medicação que ele trata a hipertensão porque ele tem uma resposta vasodilatadora. E a nossa ideia é que justamente essa vasodilatação acaba melhorando o fluxo sanguíneo e melhorando a taxa de formação óssea. Então, ele é um fármaco que ele consegue melhorar bastante esse metabolismo ósseo. A gente fez uma análise na parte medular e também na parte cortical. Mais uma vez, na parte medular, a gente observou que os resultados com relação ao SHR foram melhorados quando tratados com losartan. Uma coisa que é importante chamar a atenção é que quando a gente fez a análise no compartimento cortical, a gente não consegue mais observar tantas diferenças. E isso confirma esse nosso modelo experimental em que a gente tem que avaliar sempre a região medular, porque é só nessa região que a gente vai conseguir observar as diferenças entre os grupos. Então, a conclusão desse paper é que o losartan melhorou a osseointegração numa condição de hipertensão que prejudica a osseointegração. E a hipertensão é uma condição de risco durante o processo de osseointegração. Por isso que a gente precisa realmente ter um olhar também com muito cuidado com relação às medicações, como é que o paciente está quando ele chega para a clínica. E agora que a gente está numa outra etapa, né, no nosso laboratório, a gente trabalhou com uma série de fármacos e eu trouxe alguns aqui, a gente tem outros, mas eu quis trazer um de cada interferência. A gente agora está passando para uma etapa em que a gente está fazendo a funcionalização da superfície dos implantes. Então, a gente faz a aplicação por uma técnica que é chamada de layer by layer. Então, nessa técnica, a gente cria uma camada de revestimento com esses fármacos ao redor dos implantes. E aí, esses implantes eles têm um efeito local junto aos fármacos. Callum, we are trying now to study the local effect of these drugs. So we are functionalizing the surface of the implants. So we have the local effect of them. And these drugs, they are liberated for 14 days. This is the time that we think that is enough. Surface of the implant. So we are working in collaboration with a professor from physics. He is processing this layer-by-layer techniques. We are also doing this functionalization of biomaterials. We have some studies with biogram and other biomaterials too. But this is one of the results that we have obtained when we have alendronate on the surface of the implants. And this is a continuation of the previous study. So, essa é a continuação do estudo anterior, que é também do mesmo aluno, né? Aquele lá foi o mestrado dele, agora é o doutorado, em que a gente continua trabalhando com animais espontaneamente hipertensos. Esses são os primeiros resultados que a gente obteve no laboratório, e a gente já observou que, com o tratamento, com a funcionalização de alendronato, a gente consegue ter um torque de remoção, precisando de mais força, né, quando a gente aplicou esses implantes tratados com alendronato. A ideia é, no projeto dele, tratar com alendronato. A gente tem agora o projeto que foi aprovado pela FAPESP, em que a gente vai estar funcionalizando com teriparatida, genisteína, que é um fitoestrógeno, e uma droga que é a doadora de óxido nítrico. Então, a gente vai estar funcionalizando a superfície dos implantes com esses fármacos, numa condição de osteoporose e síndrome metabólica, para a gente ver como é que está o reparo em maxila. Tá? Então, essa é agora as próximas coisas que a gente vai estar fazendo no laboratório. Então, a gente passou por algumas coisas sistêmicas, essa administração sistêmica, e agora a gente está com um olhar para essa administração local. Até porque, sistemicamente, muitos desses fármacos precisam ser interrompidos o tratamento. A teriparatida, que é fantástica, só pode ser tomada por 2 anos. Depois disso, ela não pode mais. Então, por isso que a gente está agora com esse olhar para essa aplicação local desses fármacos. Tá? Então, em conclusão, dinâmica óssea, remodelação óssea, né, esses processos são essenciais para que a gente consiga entender essas respostas observadas nessas condições separacionais que eu falei para vocês. Importante que a gente desenvolva mais estudos, avaliando mais modelos experimentais para caracterizar o osso nessas condições sistêmicas diferentes. Os modelos em vivo, com esses estudos pré-clínicos, eles fornecem importantes informações nesse contexto, mas eles têm limitações, e isso a gente sempre precisa considerar. Cada modelo tem a sua limitação. Esse é o nosso laboratório, que a gente tem lá em Araçatuba. Essas são as pessoas com quem a gente trabalha. Eu quero aproveitar aqui para agradecer. São os alunos que eu oriento. Eu sou essa aqui que está na frente, ó, aqui. Aqui é a meninada que está trabalhando com a gente. Então, são os alunos de pós-graduação e de iniciação científica. E eu quero agradecer também todas essas instituições. Thank you very much. questions, please no.
So these implants weren't loaded, right? There was no, the implants were put in, but no, a crown was not attached.
No.
No crown.
This is another limitation. We are trying to solve this problem because we need that the load needs to be studied too.
Yeah.
For now, we just have the results, but with implants installed, and just this. Not we don't have the load. We think we are trying to solve these questions. We are talking to a professor from
Prosthetics?
Prosthetics, yes. And he is trying to develop a condition that we can put a crown, and then maybe we should study because this study that I told you when we are going to install the implants with the surface functionalized, we are going to do in molar, in maxilla. So we are trying in the next step to maybe put a crown, something like this, just to study the load applied to this bone. Okay.
Professora Okamoto, acho que eu vou falar em inglês. We have in the dentistry world, in the book of maxillofacial surgery world in particular, the loss of the osteonecrosis in the patients medicated with alendronate.
Yes.
I want to know if exists any research that have the comparison of the raloxifene in comparison with the alendronate in that case of loss.
Condition. You see osteonecrosis. Yes. What we saw in our study is that alendronate, we have a delay in the repair process, and the bone that was produced when we treat with alendronate is not a bone with good quality. So this, I think, that shows us that this drug, alendronate, is less potent than zoledronic acid, for example, that is more related to this condition. But this shows us that we need to take care when patients use this medication. And my colleagues in Araçatuba, they always show us many cases in patients that take oral biphosphonates like alendronate, and they are losing their implants. So this is a situation that we need to take care. And another thing is that the denosumab, that result that I showed you with OPG-FC, this is a trial to mimetize the denosumab. This is another drug that is also related to osteonecrosis. That's because first we use the term bronze, that is osteonecrosis associated to biphosphonates. But now we use the name bronze, osteonecrosis associated to medicaments, because of the denosumab, the Prolia, and another drugs that are anti-angiogenics. I have a lot of colleagues that study the model of osteonecrosis using zoledronic acid, and every time we see that there's a delay, a great inflammatory response that stays for all of the time that they are studying. So it's a situation that we need to take care. And just another thing that I make a lecture on Copac last month, and the theme was osteonecrosis. And I was looking for conditions of osteonecrosis associated to these drugs that we study. And teriparatide was the drug that have been used to treat the osteonecrosis. That's one thing that shows us that teriparatide is a drug that we have to look with more care.
So perhaps the teriparatide would be better than the alendronate.
Much
For
better.
not-
But the question is that, as I told, it may be used just for 2 years, because after this, they cannot use anymore. Then we need to use to keep the quantity of bone with an anti-resorptive drug. And that's the question, that we need to look for another anti-resorptive drug that can keep without causing the osteonecrosis.
Okay, thank you.
I will speak in Portuguese
Please,
because
please.
I have so many questions. so interesting. And I have some curiosities, especially because I'm not from this area. So congrats for your talk. And my first question is, o quão próximo estes resultados são de sistema humano de reparação óssea e como vocês fazem essa aproximação de sistema trabalhando com ratos para humanos, né? Já que tu falaste um pouquinho das limitações que as pesquisas com modelos têm e tal. Então, a minha curiosidade é como vocês fazem essa aproximação, enfim.
Esses estudos, esses estudos, eles já são desenvolvidos há bastante tempo, com esse modelo dos incisivos, desde a década de 60, lá em Araçatuba. Mas antes, aqueles primeiros trabalhos de Ambler foram feitos em biópsias de humanos, já se tinha uma ideia de como é que acontecia essa cronologia em humanos e mesmo as respostas também textuais. As respostas são muito parecidas. Então, a gente vê que os eventos celulares, eles acontecem mais ou menos da mesma maneira. Então, é aquela resposta inflamatória, depois tem a parte de granulação, a síntese de matriz e depois a remodelação. Isso já é universal. O que a gente tem com relação a esses animais, especificamente o rato, é que a gente tem uma aceleração dessa resposta. Então, a gente faz um cálculo, e aí vem de estudos que já são desenvolvidos lá em Araçatuba e em outros lugares também, em que a gente faz uma correlação de 3 vezes. Por exemplo, 2 meses para mim significam 6 meses... Não, 2 meses no rato significam 6 meses para mim. Então, por isso que a gente faz os nossos períodos indo até 60 dias, mais ou menos, que seria mais ou menos, no caso do implante, o tempo que a gente observa a reparação e depois de 6 meses o profissional vai lá e reabilita o paciente, dá carga, colocando a coroa. Então, é mais ou menos assim, tem limitações, mas assim, com relação às respostas são muito semelhantes, o tecido em si. E aí a gente tem essa questão do tempo.
Tá. E a outra questão também, curiosidade. É possível testar isso que vocês testam em pacientes mesmo, tendo, por exemplo, um grupo controle com diabetes e outro sem diabetes? Isso é feito?
A gente conseguiu
atualmente?
publicar um trabalho, agora, o ano passado, um trabalho que me deixou até muito orgulhosa, que a gente publicou na Clinical Implant Dentistry Related Research, que é uma revista de implantes. O Alexandre até faz parte do trabalho. E, na verdade, assim, a gente coletou biópsias dos pacientes. E era nesse modelo de hipertensão também. Só que a gente não conseguia instalar um implante no paciente hipertenso. Então, a gente fez uma coisa meio que reversa. A gente comparou biópsias de pacientes normotensos com biópsias de pacientes que eram hipertensos, mas eles eram controlados por esse fármaco. Como a gente não conseguia trabalhar especificamente só com losartan, porque tem paciente que usa captopril, e eles são da mesma classe, então a gente colocou medicações inibidoras do sistema renina-angiotensina. E aí a gente envolveu pacientes tratados com captopril e losartan, teve todos aqueles processos de exclusão e inclusão de paciente, mas tinha que ter esses 2 grupos. E aí a gente coletou as amostras no momento em que os alunos trefinavam para instalar o implante. Então, a gente tinha esse trabalho, a gente selecionou os pacientes, eles passavam por todos os critérios, daí a gente instalava os implantes e, no momento da instalação, eles coletavam com uma trefina de uns 3 milímetros mais ou menos de diâmetro, instalavam o implante e o paciente seguia para reabilitar. E aquela trefina, a gente fez umas análises tanto microtomográficas como também por imunomarcação. E aí o resultado, que foi bastante interessante, é que as características foram muito semelhantes. Então, o tratamento com losartan, ele, na verdade, assim, conseguiu fazer com que as biópsias, né, aquele tecido ósseo, ele estava muito semelhante ao que a gente observou nos pacientes normotensos. Agora, não temos nada a longo prazo, né, para ver depois, quando pôs carga nesses implantes, como é que respondeu. Essa resposta foi só assim, naquela condição inicial em que o implante foi instalado. Essa questão da carga mesmo, isso a gente não fez, eu acho que precisaríamos fazer. E a ideia é a gente, depois, começar a trabalhar com essas outras interferências também, nos pacientes. Diabetes, osteoporose, por aí vai.
Legal.
Obrigada.
Parabéns.
Obrigada.
E obrigada.
Ninguém mais.
No more question?
Então, quero agradecer a presença da professora Roberta com sua palestra. Thank you, Roberta, for coming. And this is a certificate.
Okay, so thank you for coming today. And now we finish it for today, just for today. And tomorrow we have more. Like we start at 8:30. Thank you all.